Hey now, I'm glad everybody's back. That was a very tasty lunch. Thank you for arranging that. And this is a really opportune discussion considering what we were just talking about in terms of accessibility. Why are we all doing this? We're doing this for patients and not to use the word patient as pejorative. It's actually, we're all going to be patients someday. And what would we do for ourselves? What would we do for our families? So I think altruism is kind of limited. We're trying to work on things for rare diseases, but also common diseases and rare manifestations of common diseases. So we'll start this session on patient advocacy. We're really fortunate to have heroic patients here and patient advocates, starting with Lauren Miller-Rogan, who has been on the board for CIRM for a number of years and is a filmmaker, really brings to light the importance of advocacy for patients. She's a screenwriter, director, producer, philanthropist. She has had her life touched by Alzheimer's and found herself in a position like a lot of us will, where the unexpected happens. And that's where some of us will run for cover. Other people, like Lauren, will run to be in front to lead the charge on finding better therapeutics for diseases like Alzheimer's that affected her mother. So this is a really important opportunity to talk about what we were touching on in the last session, and that's the importance of charitable organizations. So Hilarity is a charitable organization, Hilarity for Charity, that Lauren co-founded. And since then, they've brought awareness to Alzheimer's disease, raising millions of dollars for quality in-home care. And that's something that's really vital because we give people a devastating diagnosis and then we say, oh, well, you and your family can deal with this, right? And they have no resources at home. They're trying to figure out how they're going to balance their one or two jobs with this new diagnosis. It's going to take a long time for them to take care of. So this is a vital resource and a vital opportunity to hear how do we coordinate these things. The dedication, just sharing personal stories, I think is an important part of what CIRM does for patient advocacy. And I think also getting involved in reaching out to communities because you're well known yourself, Lauren, for your movie making. I think then that shows people that scientists aren't weird. Some of us are. But for the most part, we have a strong sense of purpose. We really want to help people to get better therapies, better diagnostics, and get the right therapy to the right people at the right time. But this is predicated on having patient advocacy. So thank you, Lauren, for being here. I'd like to invite you to come.
How's everybody doing? I am Lauren Miller-Rogan. I am a filmmaker, I am a CIRM board member, and I am a patient advocate. I am so very honored to be here today at the CIRM Alpha Clinic Symposium on the cusp of the 20th anniversary of the California Institute of Regenerative Medicine. 20 years, it's amazing. It is so inspiring, really, to be in a room full of people who are dedicated to changing lives by advancing science. Our home, California, as you know, is a global center of biomedical innovation, and many of you have been instrumental in driving breakthroughs forward. CIRM and the Alpha Clinics Network exists to accelerate this work, ensuring these innovations reach patients faster. Your efforts, whether in research, advocacy, caregiving, patient support, represent hope for so many people and families, ensuring that these innovations reach patients, waiting for breakthroughs that will change their futures. Personally, I know about that hope that the patients have. It's hope that I held on to when I found out that my mom, Adele, was diagnosed with early onset Alzheimer's at only 55 years old. My husband, Seth, were in our 20s at the time. We were very far from prepared. Honestly, I don't think anyone could ever be really prepared for someone you love to disappear into a disease like Alzheimer's. But we were quite young, we were scared, we were facing something that would, of course, change our lives forever. I'll never forget the first time that I decided to attend a support group for caregivers. I had finally mustered the courage to go, only to find that I was the youngest person in the room by decades. Everyone else was caring for elderly parents, spouses, while I was there because my mom, who was a teacher for 35 years, was slowly slipping away. There was no space for someone like me a younger person, still figuring out my own life while grappling with this very impossible situation. I felt so completely alone. So we formed an organization called Hilarity for Charity, which is HFC for short, let's call it now. And it's dedicated to supporting families impacted by Alzheimer's disease and related dementias, activating the next generation of dementia advocates, and being a leader in brain health education. In the 12 years since we first began, we've raised more than $23 million to advance our mission. And fortunately and unfortunately, I realized I was very far from alone. Also, a few years ago, I faced my own health scare, a cerebral aneurysm. I was diagnosed after a routine full body MRI scan and told that it was small. I don't need to worry about it as long as it doesn't grow. For a few years, of course, it did not grow. And then it did. And suddenly I was filled with fear. What would I do next? What were my options? How would this be handled? What is going to happen? But thankfully, I was in the hands of one of the best care teams in the world at UCLA, in the state that is leading the world in biomedical innovation. The whole experience was surreal, equal parts terrifying and humbling. And I remember thinking, I am so lucky to have excellent care. My experiences on both the caregiver and patient side made me realize how vital access to the right care is, to the right care truly is. Not everyone has the same access to expert care. And that's why patient access advocacy programs are so important and why CIRM is so important for funding these vital programs. In 2013, I joined CIRM, the CIRM Governing Board, the Independent Citizens Oversight Committee, as a patient advocate for Alzheimer's disease. My involvement was fueled by my belief in CIRM's mission and its drive to bring new therapies to communities across California. CIRM's funding and support are really pushing the boundaries of what's possible in science. The breakthroughs that we're seeing in cell and gene therapy hold incredible promise for the future. I can imagine a world where incurable diseases like Alzheimer's or ALS are curable. My family, like millions of others, would benefit from these breakthroughs. That's the world we're working toward, and CIRM will play a pivotal role in leading the way. It's through CIRM's Alpha Clinics network that cutting-edge therapies like cell and gene therapies actually reach the patients who need them. These clinics are among a select few in the nation, and the expertise and capacity to deliver these groundbreaking therapies. With collaborative partnerships, the Alpha Clinics are actively conducting clinical trial research for life-changing treatments for communities statewide. By focusing on patient-centered care and research, the Alpha Clinics are ensuring that clinical trials meet the highest standards while promoting accessibility and, of course, inclusivity. CIRM is now setting out to build on that commitment by creating the Community Care Centers of Excellence, a future program that will establish geographically diverse and culturally responsive centers of excellence to support access to clinical trials and approve regenerative medicine treatments. These centers will serve as a hub for expanded access to therapies, as well as education and training programs to even more communities throughout California. Further, they will develop the expertise and capacity necessary to deliver the emerging pipeline of cell and gene therapies. The expansion will enable the field to grow while improving equitable access to clinical trials and potential therapies in geographic areas that have historically faced barriers in healthcare. But having world-class clinics capable of delivering life-saving therapies is not enough. Clinical trial patients often face financial challenges, long-distance travels, and family commitments that can make it difficult to maintain participation. I can remember these challenges all too well when caring for my own mother, who participated in a clinical trial that was administered over an hour away from my parents' home. We need to make sure that Californians who need these treatments can get them regardless of who they are or their ability to pay. Proposition 14 recognized this need and established a dedicated fund to support patient participation in clinical trials and access to approved treatments. This fund will be deployed through CIRM's Patient Support Program, an initiative designed to get us one step closer to that vision. It's important to remember that programs like these don't stand alone. They are part of a larger ecosystem that CIRM has carefully and thoughtfully built. Through its work, the Apple Clinics have become a model for many of CIRM's other programs, serving as a foundation for broader initiatives like the Patient Support Program and the Community Care Centers of Excellence. These programs are critical to achieving CIRM's mission. They demonstrate CIRM's commitment to not just advancing science, but ensuring that scientific breakthroughs are accessible to everyone, no matter where they live or what their circumstances may be. And none of this can happen without collaboration. That's why we're here today. The work that all of you do, whether it's as a researcher, clinician, advocate, or someone working behind the scenes, plays a critical role in moving the needle forward. As someone who has been through the heartbreaking journey of losing a loved one to an incurable disease, I can tell you that hope is one of the most powerful tools that we have. The work being done by CIRM and the Apple Clinics give people hope. Hope that someday families like mine won't have to face the devastating reality of diseases like Alzheimer's. Thank you so much for being here, for your dedication, and for your commitment to advancing science. Know that your efforts are helping to make regenerative medicine a possibility for everyone. Thank you.
What we'll do, thank you. That was really poignant, actually. So what you can see is you can be a member of a caregiver team, but also a patient yourself. And that's the point of why we're all here, to work in lockstep with CIRM to see how we will navigate that. So for this panel, we're going to have one person online and we're going to have 3 people up here, all of whom are patient advocates. So I'd like to just give you a brief rundown of the panel and you can see who's up here. So we'll have Kim Cage. She's the director of Sisters of Hope Foundation. She's been a banker for 31 years, has worked with various financial institutions, has helped to learn and develop for businesses. Just making sure everybody's getting there. More from Morro Bay. Hi there, Kim. Nice to see you online. Thanks for joining us. She's got a family issue with ALSP, which is a microglial disorder that induces leukoencephalopathy and is driven by one mutation, CSF1R mutation. And you can be haploinsufficient for that, meaning you have 1 copy, you can have 2 copies and then have fully penetrant disease. So it's not just knowing about a family member that has it, but what do you do if you've got 1 copy, and how do you predict and prevent evolution to ALSP? So Kim will help us to understand how we lead in neuroinflammation and other diseases that are inherited, and how with ABLA's program, we may be able to eradicate these sorts of diseases that are really important. She's had End ALSP fundraisers, and then also focused on the American Brain Foundation to bring in industry partners. So thank you, Kim, for being there.
Thank you.
We'll be asking you questions as we go. Then we've got Kristen McDonald, and she's is actually the founder of Second Vision and a patient advocate. She has been an actress in Hollywood and really is going to talk about her story, having really had to deal with a rather rare eye condition called retinitis pigmentosa. I remember, Kristen, when you talked about this before and how inspiring it was to really hear your story. And I can see you're still a very strong patient advocate and are leading the charge in this disease, but also to remind us we're all going to inherit something. And having an answer comes from people like you being able to tell us how to forge the path. So you've been forging the path very well. In 2016, Kristen enrolled in a clinical trial at UC Irvine. This study used progenitor stem cells with the hope of restoring eyesight. And as a result of this stem cell therapy, now Kristen can see some light. So she's going to shed some light on what she's been going through over the last few years. She's been an inspirational speaker at the annual ISSCR conference, a keynote speaker for the US Navy, UCI Gavin Herbert Eye Institute, and she will talk about why her eyesight has been challenged. But now there's some restoration. Where do we go from here? So she's going to light the path for all of us. Thank you, Kristen McDonald. Next, we have Katie. Again, I saw Katie, as a number of us did, at an Alpha Clinic CIRM grantee meeting. I think it was at UCLA with Dr. Joe Chachi from UC San Diego also speaking on that panel. And Katie was talking about the challenges of having to decide to take a therapy that is going to be life altering after having had a life altering event. And in this case, it was spinal cord injury. She had to make the decision within 2 weeks of that injury to get an oligodendrocyte progenitor product that was developed originally by Geron, and it was the fifth and actually last person on that study. Katie has been an absolutely brave and brilliant advocate for patients with spinal cord injury. And this is a bigger issue now as we drive like crazy on the 5 to try and get back to wherever we came from. You know, there are different reasons when people get this, but I think with Katie, we're going to talk about cellular replacement therapy, but also other ways to augment or enhance engraftment from those cellular replacement therapies. Is there a neurostimulation, neurorestoration strategy that can be used? So Katie will talk about that. She has been on this journey for a little while and really led the charge. So without further ado, I'd like to go right to the questions, if that's okay with everyone on the panel. Okay. All right, yes, we will. I will sit with the panel members because it'll be cozier, and then we'll bring you in from Zoom when we get it. And the clinical journey, how did that inform your advocacy efforts and how your experiences in empowering you to support others facing similar challenges?
Thank you. Hi, everyone. I am so honored to be here again. I was actually at the very first Alpha Clinic Symposium at Stanford, I think 12 years ago now. It was exactly a year after my injection, and I had the pleasure of coming on stage and having an opportunity to speak to you all, similar to today, but about possibly entirely new things. So on the day I actually had that conversation 12 years ago, so much of what I brought up seemed to be, just baffling, right? When I told people how hard it was for my family and I to, for me to participate in the trial, to keep going back for blood work and scans, and then, eventually to do more rehab. It seemed to blow everyone's mind. People were just flabbergasted that such an experience is what we go through. And so for me to come back here 12 years on and to see the conversation shifting so much, it's very exciting. And I hope that in some way that day, my perspective and my take had something to do with all the conversations we're having, the passion that's in this room for giving more access to patients and taking care of their families and well. And that has really been, that hardship really shaped the way that I advocate for others, just like myself. I'm also a first generation immigrant, so my parents are not fluent English speakers. So just the more, attention to diversity and to be inclusive of people that don't necessarily have the support or the representation. It's been a very inspiring journey.
Oh, you're sitting right next to Lauren, who has had the prescient notion that we should have better home care. What about having translation services to go with that? Do you think that would be helpful? I mean, not that having home care isn't a great thing, but what would you add to that?
Well, I mean, I think what Hilarity for Charity does is amazing. I was actually just having a conversation with my mom last week, and I don't want to get emotional talking about it, but she was telling me about how hard it was in the beginning, especially when I was not only going through physical challenges, but mental challenges. I had a very hard time accepting and adapting to my situation. And I, unfortunately, like a lot of people, took it out on my caregivers and my family. And I know how hard it must have been. And, in retrospect too, as I'm older, I can only imagine how hard it must have been for my parents to really try to keep it together and keep me in a good place mentally. So, just seeing that there is more awareness to caregivers and their well-being as well, and just general quality of life for patients, and not just, focus on a cure of some sort, but just really giving attention to the nuances, and also how the patient's quality of life is impacted by by these things. I think that's really amazing, and I'm really excited that, this is part of a conversation now.
I am going a bit out of order because I wanted to ask Lauren along the lines of what you were just mentioning, Katie. Should we have psychological counseling for people who diagnosed with chronic diseases, especially your travels through having to deal with a brain aneurysm? So first of all, should we all have whole body MRIs? And secondly, should we have psychological counseling available for our patients in clinic?
Now it's on. Okay, now it's on. I don't think I should prescribe everyone here to go get a whole body scan. However, as someone who has done quite a deep dive into my own genetics and my own health landscape, we'll call it, I found it extremely helpful. My mom has that does have genetic biomarkers to it, and I have done such a deep dive into the hope into brain health and things that I can be active about, things that I maybe have control over that I didn't think I had control over. And so therefore, getting a full picture through things like full body scans and deep genetic testing has really helped me on my journey. But I think that the other part of your question, supporting caregivers and patients with the emotional side of things is so key. I think that families are given a diagnosis, and yes, there's the logistics of how do I find the doctor, what treatment, what trial, but the emotional load that is suddenly piled onto a family is, as you said, so huge and is of equal importance to the medical side of care. There's statistics of many caregivers of dementia patients die before the person they are caring for. And that is due to the emotional load of stress that they are carrying. And so I think that providing access to some type of emotional mental support is absolutely key, whether it's just time with the doctor administrating the therapies or a trained professional, I think is hugely important. And we see it at HFC, the programs that we provide to caregivers that are, yes, logistical, but more so emotional, are so important to helping them be better caregivers to their patient, to their loved one, but also to themselves.
And you understand the emotional journey of having had a stem cell product for Regenerative. What prompted you to become a patient? Really let all of us actually go about rare diseases. Could you have a
Okay, team effort here. Thank you so much, Kat. And I'd like first to, before I even thank CIRM and Cedars for this wonderful event, thank the lighting director, because it reminds me that I really did get a boost from the stem cell. I like to have these lights in my living room. So my patient advocacy was actually born out of being in the stem cell study 9 years ago. Prior to that, I'd always been some sort of a volunteer for fundraiser for volunteer for Center for the Partially Sighted, Foundation Fighting Blindness. I always wanted to be a part of the solution, not the problem. But it was very exciting to be a pathfinder, to be the first person in Dr. Clausen's study, because shortly thereafter, I guess someone got wind of the fact that I had, had some experience in front of the camera and liked to speak. And they said, would you like to be a patient advocate or ambassador for Americans for Cures? And one thing led to another. And that journey has been so fulfilling because it's given me the opportunity to not only engage with other patients, with other conditions and have community support, meet fascinating doctors, find out what's going on cutting edge for tomorrow, because hope is the optimum word. But it's given me an opportunity to really understand the process of what I went through and be able to talk to other families and patients who really don't have a lot of knowledge or maybe are on the fence about deciding to be in a clinical trial. I had the opportunity and blessing of speaking in Chicago this summer on a panel about being in a clinical trial. And it wasn't even until then that I realized, wow, this is my second trial. I guess I have a lot to say. So it's been a blessing, really, to be an ambassador for CIRM.
Thing you do. And then we're going to go to Kim to talk about ALSP. That's kind of leukodystrophy or leukoencephalopathy rather than CSF1 receptor driven. How has it been for you as a patient advocate, but also knowing this is an inherited disease? How have you dealt with that?
Well, first of all, thank you for having me. I'm so happy to be here with all of you, even though it's virtually.
Okay, Kim, I think you're on mute. I think this is going to be something we remember from all of our COVID, post-COVID days.
Oh, no, I have the mic on.
Hello, Kim. Can you try talking again?
Can you hear me?
Yeah, I think when I look at your microphone, it looks muted, but yeah, I see it muted.
You can't
How
hear me
about
still?
No. I'm gonna get Pablo up. Kim will come back to you, where we really want to hear what you got to say. We're going to ask you two questions at once when we get the sound fixed, okay? Thanks for your
All right.
patience. I think, yeah, we'll have to wait. Sorry about that.
All right, back to the, okay. So we'll get back to ALSP in a minute, but how do organizations like Hilarity for Charity provide crucial support for patients? And what I really wanted to ask here is, during the clinic, what patients and their families are tired of the Debbie-Downer approach? You know, that must be bad to be you. We don't want that anymore. So I'm wondering how humor plays a role in healing.
I think humor plays a huge role in healing or navigating these diagnoses that are plopped in our laps almost seemingly out of nowhere, right? And so when we first began, as I mentioned, we were very scared and we were very sad and we didn't know what to do. But then we thought, well, what is it that we do know? What do we know? Which is comedy. And my husband often jokes that if we were makers, it'd be called making for charity, not hilarity for charity. But here we are with hilarity for charity. So for us, it was about laughing in the face of darkness. And sometimes things are just so sad, you have to laugh. And I think we've used that approach in all areas of our work to infuse levity into whether it's our support groups or we just created a caregiver guide for caregivers. And we, instead of it just being this very basic PDF, it has a theme. It's a road trip. It's a caregiving road trip. And there's graphics that make it accessible, that make it not a science book, that make it from person to person, that can touch your emotions in a way that is so needed. Caregivers really need to be seen. They need to be in community with people who understand. Being alone while navigating something like this will only make it worse. And so for us, bringing humor was a way to bring people together. The first few most comedy shows, which raise money, which is lovely. But like I said, we really do try to infuse that in everything that we provide for caregivers and patients who are going through it, because for me, humor is the way that I have personally dealt with these hard parts of my life. And I've been to many lovely, beautiful Alzheimer's events, very serious and very sad. And that's great. But we wanted to do something different and to shake it up. And I really, truly have found that young people especially, they want to be loud, they want to be angry, they don't want to be polite. I always say, like, Alzheimer's is not polite to me, so why do I need to be polite to it?
Okay. So back to humor and the stay strong and carry on mentality. Which is vitally important for everyone to live with the common disease or a huge tragic outcome. When you have control over the future, you moved to London or you moved to England for a little while. How did you find culturally that was compared to being here in the US?
culturally in the sense of,
But just dealing with disability, were people different in the way they approached you? Was their response to dealing with difficult situations different?
honestly, I have a very pleasant experience, so I can't really complain too much. Anytime in my travels, I've had pretty good luck, so I don't know how much I can comment on that.
What I was actually trying to allude to, sometimes in other countries, they don't take themselves quite as seriously. And sometimes they say, hey, bad stuff happens, and you get together, galvanize to resolve, to work with family and community to have better outcomes. And then in Scotland, where my parents are from, they say, it could be worse. And I think it usually is. But it was more about how much of this is cultural, where we take on and eat our gloomy stuff.
I mean, I can't really speak to my experience in London, but I am Iranian American, so I know for me, 13 years ago when I was participating in the trial, I did have a lot of negativity from my family side, and my family was a part of the community so that they could, share events and support each other. So a lot of it was sort of directed at me, or our interactions were just directed at each other. So I did have a hard time in that sense, perhaps culturally, where and also in my culture, it's very sad when you're paralyzed in a chair. Life is really sad. Oh, I'm so sorry for you, but I'm very happy that I live in a place where I can thrive and where I have been able to become independent and do everything that I've done. So I really, I feel like in general, especially after COVID, people's minds have changed. Like people are thinking so much more about not just their own quality of life, but patients' quality of life. It's finally become a topic of conversation. So I feel like again the landscape's changing, the conversations are changing. So I'm very hopeful that even if there used to be more cultural negativity, I think that's shifting now, especially in like the last 2-3 years.
Some good things came of COVID. Okay, all right, so thank you so much, Jacqueline. So back to you, Kim. We wanted you to talk a little bit about your journey with ALSP and knowing about the CSF1 receptor mutation, where it can be genetically carried on. And what have your journey requirements been for getting on with life and doing things for patient advocacy?
Can you hear me? No?
Sorry about that. Try one more time at the podium. So used to, like everybody else, running Zoom meetings. We will see
Yeah.
about this. Okay, the mic is on. Okay, there, Kim, can you hear us?
I can hear you. but
Okay,
you all can't.
perfect.
Can you hear
Yay,
me?
good. Yeah, somehow, yes, we can hear you. I just turned the laptop way up and I'm gonna be at the podium just in case. Yeah, so we're talking about ALSP, which is a glial disorder and driven by CSF1 receptor mutation. You've been both a patient advocate, but also somebody who could be potentially affected because of transmission of that gene mutation. How has that been in terms of being a patient advocate, but also thinking, wait a minute, this could be me someday? How has that gone?
Well, thank you for your question and thank you for having me. Yes, ALSP came into my family's life in 2019 by way of my youngest brother. He was impacted first. It impacted his motor skills, slurred speech, and within about a year, he was unable to walk. My middle brother and I were tested for the gene mutation because we wanted to be donors for my youngest brother. A bone marrow transplant was an option and could potentially stop progression of the disease, and it has done that. But in the midst of all of that, my middle brother Joseph and I had confirmed diagnosis for the mutation. Since then, my middle brother started having cognitive issues and was confirmed to have ALSP in 2022. He is living the remainder of his life in a care facility. And as you stated, I have the gene mutation. So my family's unique in that we have one who is impacted physically with his motor skills, the other who is impacted cognitively, and I'm still asymptomatic. I joined the Sisters Hope Foundation, the only organization that supports families and patients with ALSP, and I am a patient advocate for them. I became excited to do that after ALSP first hit my youngest brother, because I really believe in awareness, especially for rare diseases such as this. No one had heard about it. I've spoken to in many meetings, whether it's chamber events or a room filled with neurologists, and they always walk away saying, I've never heard of this disease. So it's so important to get the word out. Genetic testing is very important. And part of my journey is encouraging people to get genetically tested. We did figure that my father's side of the family is where this disease came from through my grandfather. And I've had several meetings with my cousins on that side of the family. But the thing that I hear most often is, I feel fine. But the problem with that is that you can't feel brain changes. So some people are skeptical or fearful to get genetically tested because there isn't a cure and there aren't any FDA treatments out there. But there is hope through organizations like CIRM and other organizations that Sisters Hope Foundation partners with, who are working toward stem cell therapies. And there's also, opportunities for gene editing through CRISPR. So we hold on to hope, and the journey is just beginning for me, as far as I'm concerned, as far as raising awareness and really building this journey with the Sisters Hope Foundation. And so I'm very happy to be here and be able to talk a little bit about ALSP.
So I had a question. Sorry.
If I may
Yes.
one moment for some baby wizardry. Excuse me. We're going to try and reroute this so our virtual participants can also hear us. Booth? Yeah. All right, Kim, can you try talking one more time?
Yes, can you hear me?
All right, give us this. I promise this is going to be wonderful.
Okay, go ahead, Kim.
Can you hear me?
Remove the stigma. When we had pre-existing diagnosis, wasn't just a stigma.
This all the way down. Yes.
I know that there was one I tried, I was doing three things, muting,
How do we move to make sure that that's part of regular healthcare? Is there a way to advocate for that at the state level, federal level? How would you say that charitable organizations play a role in that?
Are you speaking in regards to the genetic testing?
Kim, can you say that again?
Can you hear me?
It's too hard.
Okay, Kim, can you hear us now?
I can hear you.
How about now? Is that good?
I can hear you.
Okay. All right, can everybody hear Kim now? Yes.
Here.
Okay, Kim. Sorry. I think. Okay. Gonna do one thing, just a sec. All right, how's that?
I can hear you all perfectly.
Yeah, so I think, oh, we're gonna have an echo. I think what we're trying, I'm just gonna let you say a little bit more about the road to advocacy for genetic
Here, let me
testing
I'll
and
go down.
clinical trials.
You got it right. Just
Okay, can you hear me?
Yes.
Okay. As far as genetic testing goes, I think it's important for people to understand, at least for the ALSP community, is that we do offer free genetic testing through Sisters Hope Foundation, and we do that in partnerships with other organizations. I have often heard people say, well, it's too expensive, or my neurologist told me that I don't really need that. I've been diagnosed with this particular disease, and they're certain that that's what it is. But I think that we need to really encourage people to understand the benefits of getting tested. ALSP cross-references many other diseases as far as symptoms go. So it's really critical to be able to understand what the symptoms are, get genetically tested, and confirm that CSF1R gene mutation. My youngest brother was misdiagnosed for 2 years with multiple sclerosis, and it wasn't until a neurologist out of UCSF saw him with his neurologist and said, this doesn't look like MS. We need to get a genetics test done. So I think it's important for neurologists especially to be aware of this disease and really push for genetic testing. I can't hear anything on my end.
Getting back to the alpha clinics, if that's where we're here. So we have 9 alpha clinics in the state. We see all the directors here. We work very collaboratively to implement cell and gene therapies, but also small molecules and biologics that enhance endogenous repair and regeneration that can be partnered with these therapies. How would you see the role for the alpha clinics in terms of retinitis pigmentosa, other forms of blindness? What are your thoughts?
I would think that some of these other organizations that I mentioned in the past that relate to having an eye disease, such as Foundation Fighting Blindness, Braille Institute, and many others that I've connected with over the years, I would think that it would be a great thing to have some sort of collaborative effort on some level. You know, information, you know, when you first get diagnosed, you're in the, you might be able to see that you're in the dark, you know, about this world of blindness. It doesn't come with an instruction booklet. It comes with a lot of training and independent living skills. And there's a lot of resistance to that sort of thing, psychological counseling, et cetera, et cetera. But most of the doctors are just short on information in that regard. And I would like to see more of a collaborative effort with the clinics, you know, for information, at least at best, maybe providing even a volunteer network. There's so many people who are visually impaired who it's not only till, I don't know how many number of years now, 6, 7 years that I've been really speedy at doing my own mail because of the technology with voiceover. But in the past, I always had volunteers and people need help. They get isolated. They need to get out more and more support, I would say, community support.
The alpha clinics play a role in that education, Lauren?
When I first started talking about Alzheimer's and dementia, I felt that there was no hope. And, you know, this was 15 years ago and treatments were not where they are today. And the words brain health were certainly not a part of the conversation. And I think that part of the reason people are scared to get genetic testing when they have a family member with dementia or Alzheimer's is because they feel like there is no hope. But the truth is today, that's no longer true. We can do things to keep our brains healthy. HFC created brain health coursework for people to teach them how to keep their brains healthy. Science tells us that 4 out of 10 cases may be preventable or delayable, which is huge. And if we can teach people through these clinics, through outreach, how to live a brain healthy lifestyle, how to sleep better, how to eat better, how important exercise is, mental fitness, emotional well-being, then perhaps someone's dementia, perhaps their genetic roadmap that they were born with could take a detour. It could take them longer, and science could come up with a cure. And so I think that teaching people that there is hope, there is still fear, of course, but within that, there is something to be done. There is something you can do. And I think that sharing that with everyone, whether it be with a family who has a member diagnosed or any young person who has a brain, which is and they say half the country these days. And um, but that I believe is so huge.
Right, so back to Katie, I think that's a great point. To get back to this idea of, or, you were very brave to make a decision to have early intervention after spinal cord injury as opposed to waiting for the inevitable. What are your thoughts about not just how delayable some of these diseases are, but how you educate all of us to think about taking an early action? How would you get rid of that fear, knowing they're making big decisions, but what role do patient advocates have to play? As we were hearing from Kristen and Warren about clinical trials and how to educate them.
I think one thing that's very important, I know for me, when I was making my decision to participate, one of the things that really helped me was to speak to the person who had the injection prior to me, I think maybe just a month before. And it just sort of, I mean, neither of us knew what was in store for us, but just knowing that there was someone else that went through just what I was about to go through and that he was fine, he was there speaking to me. It just gave me a lot of, that I felt a lot more comfortable with my decision. And so I feel like one of the things that, I hope comes out of this, like speaking of support, speaking of providing that kind of network for patients and their families, I think it's really important to put participants, somehow in touch with each other. And I always say, this has nothing to do with the clinical trial, but I always say that in regards to my spinal cord injury, everything I know how to do, I know from learning from my peers. Truly, and I don't think I learned that much in rehab. Everything from the most basic human tasks you can imagine, I've learned from other people with spinal cord injuries. And so I feel like it's so important to have that connection and that network, those sharing those kinds of resources with one another of someone who's been exactly through what you've been through. I think that's very, very important.
A patient like me portal on a health clinic sponsored network where everybody can help you.
I think that would be amazing. I'd love something like that. I'd love something like that for me. I'm still in touch with that person who had the injection prior to me, but I'm very interested to know how the first person's doing. I haven't spoken to them in probably 11 years. So, I think that would be incredible.
Like,
Just make community, like community is key. As I spoke, I mentioned that I felt so alone, but of course realized I was far from alone. And I think that when you connect with someone who has walked in your shoes, to feel seen, to feel heard, gives you the energy to keep going.
energy, how you kept going after seeing this glimmer of hope from your vision starting to come back, knowing that it didn't entirely come back. What are your thoughts about future augmentation of some replacement therapy through devices or new small molecules? What are your thoughts?
I am hoping that stem cells will either marry gene therapy or I'll be available for a gene therapy study coming soon. I've been waiting decades for things like stem cell and gene therapy were only something that we talked about years and years ago. It was like science fiction. And now it's here. And just having gone to the Visions Conference 2024 this summer, there's 50 trials for RP going up across the country for gene therapy. And some of these are non-gene specific. So I'm very excited because just to quote an article in the Washington Post recently, they had 3 totally blind people who after gene therapy, they were at late stages, are now able to cross the street. So I'm very hopeful that they'll either work together with the stem cell, but that I will be one of those participants. And I'm still grateful for the lighting in here.
So Pablo, maybe you can help us online there. Just to follow up on that idea of gene therapy, how do we make this more accessible and affordable and maybe have an economy of scale where we find out more about the burden of disease in the community by getting genetic testing? What are your thoughts about making genetic testing cheaper, more available to get into these very, erudite gene therapy studies?
Can you hear me?
Yes, we can.
Feedback there. Can you please repeat the question? It's kind of hard to hear. There's a lot of echoing going on.
You know what we're going to do? We're going to let you just tell a little bit more about your journey because there's feedback if I ask questions.
Can you hear me? Okay, great. I think part of what I did hear was more about the genetic testing, I believe. And again, I can't stress enough how important that is, at least within the ALSP community. As far as patient advocacy goes and the patients that I speak to, they remain hopeful for some kind of stem cell therapy. I think everybody does across the board. And we know that, there can be challenges that we face with trying to get things approved through the FDA. But that's the thing that we hold on to. And as far as the patients go, we have patients, across the spectrum. These patients are getting hit at the prime of their lives, whether it's their late 20s or late 40s, and they are not mentally or financially prepared for a devastating disease like this. So as we continue to spread the word, raise awareness, and partner with organizations that are willing to listen to patients, we get more and more excited about the work that's being done. And we know that they're listening, the researchers and the scientists, they're learning more from our patients, and the partnership is going to be key. It's just a huge collaborative. And so we look forward to our continued partnership with CIRM and with organizations like Vigil Neuroscience, as well as Savannah Biotech, which is formerly Novoglia. And again, genetic testing across the board, everybody should know that at least for ALSP, Sisters Hope Foundation does offer free genetic testing, and it's very, very important.
What do the alpha clinics at CIRM still need to do?
Well, my hope for the future, just to reiterate, is that I will be a lucky participant to be able to participate in the third trial, and many people never even participated in one. Years ago, I had a genetically modified cell put into my eyes. I guess it was 16, 17 years ago, and it unfortunately didn't work. But my second trial, as you know, I got some improvement. My third, 3 times the charm, I believe. So that's my hope for the future. And I just am so grateful for the work that everyone in this room is doing, to not only, have patient advocates like ourselves out there as a voice, to see what's important to patients and doctors, but that they're working and it's their relentless quest for a cure for conditions that we all are challenged by. So many different conditions. So I'm hoping just for more collaboration with some of the charities that we talked about. And by the way, if anybody wants to do a great beach walk tomorrow, the Foundation Fighting Blindness is doing their annual vision walk. So just speak to Jacqueline and myself if you'd like to stroll along.
Nice stroll. So Lauren, as a patient advocate, you were involved in helping other people, and then you found your own issues. Is that sort of altruism something that we should all take on?
Yeah, I think that we're not doing something that is outside of what we know. And I think that we are doing what we can do. Not everyone can do the same thing. And I think that it's important for each individual, each family to sit back and say, this is what I'm able to do. I may not be able to start a huge organization, but I might be able to attend a walk. I might be able to share my story. I think that the more people recognize that every individual has the power to do something, even just sharing your story will move the needle. And so my hope in the future is that more and more people come forward and share how important it is to talk about these diseases, because talking about it leads to change. Like I said, when I first started talking about this in 2009, the conversation was pretty quiet. It now feels much louder. And that's because people have talked about it. There has been more funding towards research and science, so much more from 10 years ago, 12 years ago. And that is due to a need which has stemmed from, I believe, patient advocacy.
So final word, Katie, patient advocacy, advancing science to the clinic with alacrity. What's your hope for the future as somebody who's been a leader in getting us to get off the couch and do something good?
I think very similar to what was mentioned by Kristen. I would really love more collaboration. What I mentioned about putting more participants in touch with each other, creating those sorts of networks. But another hope of mine is to really leverage technology and advances in technology to, marry that with stem cell and gene therapy and see where that takes us. Because I've been saying this ever since I got injured. I really wanted at the time for people to start thinking of these things together. And I finally feel like we are, and that gives me a lot of hope. And I feel like we're already going in that really great direction that I was hoping for many years ago with the creation of the CCCs, the Community Care Centers for Excellence, the Patient Support Program. These are all things that we're working towards that are going to be realizing the things that I was dreaming about 13 years ago when I was hoping to have someone be there for me or someone to figure out, my Medicare coverage analysis. Like I just hearing about these things, it blows my mind now. So I'm so happy. I feel like we're already moving in the right direction, and it's so exciting and inspiring for me. And I know that those of us with chronic conditions or illnesses, diseases, there's a bright future ahead, and I'm very happy to be part of the conversation.
On that note, I'd like to thank our panelists for being the brave advocates for patients.
True privilege to have two invited speakers, both of them from outside California, both of them speaking and guiding the next generations of research and of clinical trials. I'll start with introducing Dr. Marty Gomberg-Maitland. And she's a cardiologist and a professor of medicine at George Washington School of Medicine and Health Science. She is the Walter G. Ross Endowed Chair for Clinical Research and the Chief Clinical Research Officer for the School of Medicine Health Science Office of Clinical Research. She is a very well-recognized expert on pulmonary hypertension and heart failure, with a translational research focus with over 150 manuscripts, with the most recent one on New England Journal of Medicine. Her work often explores novel therapeutics and optimization of existing treatments to improve patients' outcomes. The second speaker is Dr. Stephen O. And Dr. O is the Deputy Director of the Office of Cellular Therapy and Human Tissues in the CBER at the FDA. He oversees product quality reviews, policy development activities, and regulatory science research programs focused on cellular therapy and tissue engineered products. He also heads the bioinformatics regulatory group review in CBER. Dr. O has been with CBER for 17 years and has served in the past as a director and deputy director of the Division of Cellular and Gene Therapies and the Chief of Cell Therapy branch. Even though we have been talking about the FDA behind the FDA's back for the last day and a half, we will recognize and embrace Dr. O, as he has served on the faculty on the Tufts University School of Medicine. He is trained as a cell biologist and biochemist at University of Michigan, Johns Hopkins School of Medicine, and MIT. So let's start this exciting session with Dr. Gomberg.
these are my disclosures, which aren't really relevant to this talk, but I should say that my husband is employee right now, wasn't until young. So I do have some understanding of all of this research that everyone's doing. And I wanted to start by saying, I'm gonna try the computer. Guys, I'm okay, that works with me. All right, maybe if I angle it the right way. So why me? You know, I'm not a regenerative medicine or genetic specialist. From the East Coast. Actually, my dad grew up across from Yankee Stadium, so keeping a low profile today. Um, I'm a little bit scared, but I've been keeping quiet. But I do clinical trials, and I specialize in the treatment of rare disease pulmonary hypertension. For those of you in the audience who are not clinicians, it's high pressure in the lungs. Pulmonary arterial hypertension is complex, rare, fatal disease that when I started 20 years ago had limited options for treatment. Early studies were really straightforward. It was randomized, double-blind, placebo-controlled studies. We didn't have any therapies, very easy to show a difference. But now I think what's really important is that challenges faced in a rare disease can help clinical trials throughout medicine, irrespective of what you're trying to show. So a decade later, we're faced with many challenges, and there's a clear need for innovative trial design and endpoints, a need to demonstrate additive benefit. Patients are a scarce resource. I think that holds true for all of medicine, not just in rare disease. There's a lack of validated biomarkers in pulmonary vascular disease, and we're not learning enough about the mechanism, the dose, and the medication interactions in phase 1. And I think all of this applies for your research. Clinical trial efficiency, that's what we need. And it's really a balance between the research value, the health impact, and the validity to the research costs, which we heard a bit about this morning. You know, how are we going to be able to afford to give these trials? And it's crazy how much it costs to develop a drug. And as we all know in this room, many of them fail, or the majority of them fail. And I'm also going to touch a little bit on the key opportunities for artificial intelligence to try to improve trial efficiency, which I think was one of the questions earlier this morning. So I want to start by saying that phase 1 learning studies are really just that much more important. I look through all the CIRM studies and see that you guys are covering a host of topics and diseases. And the majority of the studies you guys are doing are phase 1. There are some phase 2 dose escalation studies for safety, but I think it's really going to be important to sort of think outside the box and see how we can strategize to be more efficient. So how can we do this better? I think learning is key. You know, we really want to get the most knowledge from the fewest patients and place them at least risk. So I think it's really important to keep an open mind. Sometimes unexpected events occur, and we know that our preclinical is pretty good. I can tell you in pulmonary vascular disease, we've cured pulmonary hypertension in the rats and the mice, but it doesn't always work when we give it in phase 1. And we really need to learn because there are some things that we just don't expect. There are downstream activity that's happening. There's off-target effects. We need to really learn that as much as we can early on. It's also really important to appreciate that pre-treatment regimens are different. There's different absorption. The mode of administration and the concomitant therapies all play a role in how the patients are going to respond to therapies. And we really need to gather as much data to determine mechanistic benefit. So how can we do phase 2 better? You know, we really want to streamline the operations to provide the patients the opportunity to participate. We need to continue to learn and really have a defined population. You need the ability to adapt, which you're going to hear a bit about in the next few slides. And we need to really work with our statisticians to creatively design novel methodologies and novel endpoints. So I know that you guys have been discussing this a bit over the last 2 days. master protocols and platform trials. And this was a nice review in the New England Journal. I think it's important to differentiate between umbrella, basket, and platform. So umbrella, you have multiple targeted therapies in the context of one disease versus a basket where you have one targeted therapy in the context of multiple diseases, and then the platform, which is multiple therapies in the context of one disease, but you keep sort of altering the study as it goes based on the results that you get, so that you can continue to enroll. And we're going to get into a little bit detail. This is a slide from one of our latest papers. So I've been fortunate to be involved in the World Symposium on Clinical Trial Design for the last decade, and that's how I met Dr. Luce here. It's been phenomenal to see the growth in pulmonary vascular disease. And what you can see here is that it's really how are we going to do a platform trial? You know, it doesn't matter what the disease state is, but it's a standard of care. You have 2 treatments. You do an interim analysis, and you know what? If one of the treatment doesn't work, you just drop it and you keep going. But it's also nice because you have the flexibility to add a different treatment and keep going till you get efficacy. The advantage is that it streamlines the operations. It's a centralized shared governance and the shared controls, which I think is really key, because it's nice if you have similar protocols with similar inclusion, exclusion criteria so that patients can jump from one trial to the next. This clearly would improve the efficiency. The disadvantage is that you need to really plan and coordinate. You need to have, there's going to be changes in standard of care during the course of studies. There are multiple routes of administration and placebo challenges. It's a much longer duration. There's funding challenges. And what we find in pulmonary vascular disease, which is a small disease state as far as comparative to my cardiovascular clinical trials and acute MI, there's a perceived risk for industry and data quality and data sharing. And it's, how do you get over that hump so that everybody wants to participate? For them, it could be a very fast way to know that your drug doesn't work, but they don't look at it from that perspective, unfortunately. Moving on to adaptive trial design, adaptive is used a lot in our terminology, and really it just means that we can change anything in the study. So you can adapt the patient number, the study duration, the endpoint selection, patient population, the number of treatments. You can drop an arm, you can add an arm, the interim analyses, and your hypotheses. The problem is that you have to unblind at some point, and then you're going to lose statistical power. So we've been using adaptive enrichment strategies in pulmonary vascular disease, both in enrollment and in futility. So, in enrollment, it's maybe we want to enroll a certain type, a certain classification, a higher risk, versus futility, which, hey, maybe the interim analysis will tell us this is not something that we want to continue in, or that we need to alter the sample size. We can enrich on the phenotype. It would be nice if we knew which patients were more likely to respond, and prognostically. And that's what we've also been moving towards, risk scores. So over the last 15 years, we've developed risk scores in the disease state, and maybe we just need to enroll folks that are at a higher risk for events so that we can have a faster trial and move it forward. So this is a nice schematic, just really showing you that you have the full cohort, you have an interim analysis, you determine the greatest benefit in which subjects, whether it's through a biomarker or some other endpoint, and then you're able to enrich your population. And below you can see the example where you're using the biomarker, you're testing 2 different treatments, you do the interim analysis, and then you can enrich for both treatments at the same time. Again, you're just trying to be smarter in the design. This is a trial that we just, the model that we just completed actually in pulmonary hypertension, which I think you'll start to see more of. It's a phase 2/3 adaptive design so that you don't run into that problem where you have phase 2 and then you analyze the results and then you have to restart again and motivate to do the phase 3 and get all of your resources together and start recontracting. And so it's a good thing. It reduces cost and study duration. There's a smaller sample size, and it works well for repurposed drugs and for, I think it would work well for the cell therapy trials. The weakness is the interim analysis, losing the statistical power. You really need to have some understanding of what the benefit's going to be, and there's always statistical issues that you can't always overcome, which is the study-wise type 1 error. But fundamentally, what was sold to all of us was this is seamless. And it's seamless for the patients, which I think is a big benefit, but it really wasn't seamless on the operational side. It was very difficult to get our clinical trials offices to understand how this was a 2 and then a 2B, and then some of it would go to 3 and the different consent forms. So I just want to put up that warning sign that it's not as seamless as one would think. I think we're going to get better at it. But what did occur was that we had a PH study that started with 3 doses versus placebo. And actually, when they did the interim analysis, none of the doses were effective. So they were able to just close it. We were ready to go for phase 3, but I think they just underdosed, to be honest with you. And again, that pointed us to the phase 1 that we did not do and went right to phase 2. So I think it's really important to learn as much as you can early on. This is my favorite, which is a randomized withdrawal, which you do a lot of in oncology trials. We hadn't had the guts to do it in pulmonary hypertension because folks were always worried about giving medication and taking it away. But you give the medicine, you have a run-in period. So now patients that don't tolerate the medicine, they're out. Patients that are super responders, also, you don't want to be randomizing them. You want to randomize the folks that have some response, tolerate the medication, but it's not a blockbuster response. And then you're able to randomize and truly see how patients are doing on the treatment versus placebo. The strengths to this is it really minimizes drug exposure. And the folks that don't respond or have toxicity are not enrolled in the study. And you can test a bunch of combinations. And frankly, we don't know, just like in cancer and other groups of diseases, we are on a lot of background therapy. Do we need to be on all this background therapy? And how long do we need to keep it? So I wrote a few examples. One would be somebody who's on dual or triple therapy for whatever the disease state, and you basically randomized to 2 or 3 therapies versus placebo. And you know what? If they develop clinical symptoms, you resume the therapy. So that would tell us that actually these patients are responsive and need all the medicines. It's a little bit, the safety behind it is going to be dependent on the disease state, but it's a worthwhile endeavor. The second would be, you have an open label extension study, and then you randomize to either you continue the therapy, which tends to be safety, versus a down titration of medication. And I think that's also nice. And then I found this, which was a cell therapy example. It's patients who had renal transplant and received bone marrow derived mesenchymal stromal cells, and they were randomized to either early TAC withdrawal or just continuing the tacrolimus. And for the background, tacrolimus has cardiovascular effects, and we're trying to see if maybe we can minimize that. And it was a very interesting study in that they found that it was safe to administer the bone marrow derived stromal cells, but they were also able to discontinue the tacrolimus. And I think that came up today too, of sort of, can we be on less chemo or other therapies while we're doing our cell therapy? And they found that they also got favorable cardiovascular outcomes, which they hadn't expected. And so I think that this is a good use of a randomized withdrawal trial. Of course, with anything, you might now be sub-selecting and you might overestimate benefit, but I still think it's something that's worth looking at. Moving on to artificial intelligence, we talked a bit, I think, this morning about technology and how that's going to help us. We didn't really touch upon how we can use it for screening and be more efficient in using our EMR. We can use it for real-time patient reporting data. I think that that's really important to get what the patients are feeling, and objective data with things like wearable devices and activity monitoring, and even implantable devices. We can help to improve subject retention. We can predict things. We can predict dropouts. We might even be able to do a better job of endpoint analysis by using technology. You can look at things at high volume. We're looking at EKGs in cardiology, stuff that you wouldn't normally pick up. Now we can do it in magnitudes of volume to really get precise predictions. And then this was really kind of a neat thing, which was machine learning or phenomapping. And you were basically looking back at studies, and what they showed was that you could actually learn while you're doing the study to figure out what phenotype's responding, and then just enroll that phenotype. So I think we need to use our technology worse in a more organized way so that we can get to the answers faster. This was also something that was new to me, which was a digital twin. You can make a patient's digital twin. And really, you're using a lot of, I took statistics and clinical trial design and such. And I always was told when you got to neural networks that you really wanted your statistician involved. And that's what I think of when I think of this digital twin, because you're really using statistics and mechanistic modeling to really figure out in the individual patient their twin. And so what you can see is that you're making predictions of how the disease is going to respond, and you're going to need to validate these predictions to make sure that they're actually real predictions. And so it's kind of taking it to the next level. They're doing this in cardiology. This was a paper from 2020. And what you see is there's a whole host of ways you're getting the clinical data, which we're all familiar with, omics and reports and experimental recordings and imaging in cardiology, and even just going to the patient themselves, like mobile health clinics and monitors. And you can see the mechanistic models, statistical models, deductive and inductive reasoning to get better results to make predictions. And I think you still need the clinician, and you need the clinician to teach to get these AI algorithms, but I think it's going to be a way to really improve what we're doing. I wanted to just quickly touch on some composite endpoints. I know that Mike Farkey talked about win statistics. Now, win statistics, we're using a lot in cardiovascular medicine. It's an alternative approach to positive endpoints by actually ranking them and what's important, and then looking to see how many of them have a win ratio versus win odds and net benefit. And so the easy way to explain this is on this graph. You have 100 in each arm, one in treatment and one in control, which then gives you 10,000 pairs of patients. And then you see how many you're predicting, how many wins, how many ties, how many losses. And you're figuring out the ratio and the odds of winning and the net benefit. You're almost like you're in Las Vegas, but in clinical trials. I think it's something, say again? No, I think it's something that's, at least it's showing meaningful benefit by prioritizing, which ones you think have the most value. It's kind of like that weighted composite, but in a different form. It's the importance of the severity of the components in the composite is important. And what we did in pulmonary vascular disease is we actually did a meta-analysis and looked to see how we did as far as in the win ratio, win odds, that benefit, and we showed improvement in all of these things. And so, it's not a panacea. It's still driven by the most frequent components. It's sensitive to follow-up time and centering. And we're really not sure what to do with it. I'll say that I'm not as excited about it, but I do think that there's a role for it. I am more excited about recurrent events as an endpoint because I think it's really important to the patient, and it's going to increase your power, and it allows for something that's clinically meaningful. But again, you have to realize that it's the same patient having the events, so that involves more statistical modeling. And then I just wanted to end on trial diversity because we've heard a little bit about that in multiple talks today, but it's really something that we need to do, and we have been doing a really good job with. It's not just to improve representatives in clinical studies, but it's really also to support more equitable and timely access, which we have been hearing about. It's going to make it more generalizable, will improve our understanding of disease in all patients and inform on safety and efficacy. This is pulmonary vascular disease studies. And what you need to know is that the orange, yellow, and green bars are underrepresented populations. And you can see we're just doing an awful job. And I think we're doing an awful job in all clinical research because of all of these reasons. Social determinants of health are variable. There's a lack of trust in the medical system, which we heard this morning from Cedars-Sinai on how to get that community engaged and to build trust. We need to use technology to increase the imbalance. So I know that we tried to do a smartphone AFib study and we couldn't enroll anybody because nobody had a smartphone. So it's like, oh, we're being really creative, but we're not getting that diversity and really getting stuff that everybody can utilize and get involved in. We need to improve our communication, education. There's a lack of diversity in principal investigators and in the clinical trial staff. And we need to really understand why we're not enrolling. And then the protocol itself, right? We all know and we heard about the visit burden and missing work and caregiver time and transportation and health literacy, very demographic distribution at different sites and how we're getting materials to folks. I wanted to just say that the FDA does now have one of its methods that it's going to do is have this guidance on diversity action plans. They're going to have action plans that are going to be required for all phase 3 so that sponsors have to say, this is our goal and this is how we're going to get to our goal. I think it's a good first step in trying to improve diversity. And then this is being done by folks in Northern California in collaboration with Morehouse. And what do clinical trialists want to do? We want to study why things are happening. So this is 2 studies actually trying to study why we are not able to enroll. So you can see on the bottom the different things that people are including. We heard about today and trying to figure out how to better do this. The only way we can do that is to study why we're not succeeding. And so I just wanted to conclude that we can do better. Early in development, we need to be learning because I think that's really the critical part of your study. It's imperative to report trials that were not successful. I think we don't do a good job of that in medicine all around. And I think that this community is great because you guys are all communicating with each other, good and bad. And not failure isn't necessarily failure. Sometimes it's just not working in that setting. And it's always good to learn from things that may not have been a success so that we can do better the next time. Collaboration and creativity is needed. Technology, I think we need to use that more in our methodology and approach. And we obviously need more diversity and enrollment. And with that, I thank you for your attention.
There's something I need to do. That's not, did I do that? I don't know what I'm supposed to do. Oh, there we go. Okay, thank you. So, my name is Steve No. I'm here today to talk a little bit about how the cell-based therapies are developed and how all these work in terms of when you
actually get to manufacturing as part of the clinical development, how it works, and talk a little bit about different ways of manufacturing products, such as point of care manufacturing. So I'll get to all that. My hope is that I'll give some regulatory advice, considerations, I should say, that would really help those who are interested in trying to come up with a strategy that would align the product development with the clinical development program for cell-based therapy products. So let me get to the next slide. So development of, okay, perfect. Development of cell-based therapy products, and I'm going to interchangeably use cell therapy, cell-based therapy, cell and gene therapy. They all mean the same thing, as long as there's ex vivo cell modification involved in gene therapy as well. So I'm going to be interchangeably using those terms. So for the development of cell-based therapy to happen, we have to recognize that there's a different paradigm that we need to work under. And shown here is an example of a manufacturing paradigm shift. So if you look at the left side, the conventional drug biologic, you will see that with one product lot, you will be generating multiple doses for many patients. That's just not going to happen for cell and gene therapy products unless you have a really well-defined master cell bank that will last you forever. I mean, that could happen too, but in typical cases, most of the product development will face something that you see on the right side, that the manufacturing of the product is going to be limited to just few doses. And that's going to be the main challenge as to how we're going to solve about this. It has everything to do with a high cost. We talked about a lot of other things that cell and gene therapies have the uniqueness in terms of not only how the trials are designed, but how it is manufactured. It all becomes a one big challenge. So when you look at some of these unique challenges, people have thought ways to go about it, and that's using things like advanced manufacturing technologies and analytical technologies. You know that that manufacturing these cell and gene therapy products is very complicated, and in terms of the process and the distribution and logistics is really a nightmare. It's very challenging. However, you know, with these challenges, people do have ways to think about alternatives, and one of those are distributed manufacturing and point of care manufacturing. Those are not necessarily the new novel concept. It might have been around for a while, but it never really saw a time where it could really be used in a more dramatic way. And we're starting to see the use of this other types of manufacturing strategies in this space. So I'm not going to go through all of these slides, but the point that I want to make here is that the clinical, while the clinical program is really progressing rapidly for many different reasons for cell and gene therapy products, it could be that just that the product is so good that you know, you have such dramatic effect in small size phase one, and that's just how it's gonna how the product is gonna be developed. It could be that that you get a lot of these expedited programs being out there for product development, for clinical development, I should say, like RMET designations, breakthrough designations, and fast track. That that just the clinical development is going to just go very rapidly. Now the problem is the product development or the CMC development needs to happen is really not catching up in a lot of the cases. And that's where I wanted to talk a little bit more about today, how to really think about bringing the product development in line with the clinical development program itself. So two are aligned in a way that it moves in parallel, and that you get to the goal line more efficiently, more quickly in that way. So the problem with the manufacturing cell and gene therapy product is that unlike any other biologics that we have seen in the past, or the small molecules, that there's so much unknown, as you can imagine. We only know what we can measure, and that's just a little bit of everything that we that could be going on in the cells, and how we have dealt with is that we set a type of critical quality attributes that we want to study, and from the regulatory point of view, that those that we hope to think of as a critical quality attributes are identified in a way that it will define the quality of the product, and it will speak to the potency of the product as it gets manufactured lot after lot. So that's the kind of approach we've taken, and as I've shown here, again, I'm not going to go through everything here, but by end of phase 2 or before the initiation of phase 3, you would at least want to have a decent potency assay in place. Potency assay that would really be able to distinguish good product from bad product in terms of when you manufacture it. And then you would want to have a lot of product characterized so that it can be used for other types of purposes in understanding what is really driving the therapy, and when there's manufacturing change, with these additional characteristics of the cells that you now understand, that you have identified, will help you to support that, yes, the new process that you're making change to is really providing the evidence that it really is not the same, but it is close enough. And that kind of evidence can only come from additional characteristics that you may not be measuring for your lot release assay. So that's sort of where we are with this. So I think I already talked about the early product characterization, the importance of that. Why do we want to do early product characterization when the clinical trial is really going on well? There's really no time and effort that we could put in for additional product characterization. Yes and no. If you put some effort in early product development, there's definitely a benefit at the end of the road, because whether you like it or not, when you manufacture, there has to be time when you would want to make manufacturing changes. Again, unlike little small molecules where you can measure just about everything, there's no way we can do that with the cells. Then again, it comes down to, do you have a data that shows that product is comparable? And the only way to do that is that you have data, and then to have the data, you need to have an assay to do that. So that really pays off at the end. And I just wanted to stress that here in this slide. Talked about a little bit about comparability already, but here, if you want to have a global strategy in terms of product development, again, you'll come to a place where you will need to do a comparability exercise some point in the product life cycle. And these are some of the things you would want to consider when you design, when you strategize product development that moves along with the clinical program. So the best way to do comparability exercise is using head-to-head comparison or side-by-side comparison of the old process and the new process. And if you don't have the old process still in place, in other words, if you already retired the old process, there's no way you can do that side-by-side comparison. Now, at that point, the only thing that one can do are two things. One is take out the retention sample, an old retention sample from 10 years ago, use that and compare with the product that you make with the new process. Or you have your historical data from the previous manufacturing, and you compare that with the new manufacturing process and try to make some judgment on whether the product is comparable or not. That's sometimes fine. A lot of times that has limitations. So I just wanted to make a point that when you're strategizing how to develop products, you try to use manufacturing process that will last you for a long time. But if you cannot, don't retire the old process, if that is at all possible. Sometimes I understand that's not possible. So I just wanted to mention that to you. I think that's the last slide in terms of the product development part. Now I'm going to spend the rest of the time talking about 4 different manufacturing strategies. And those are centralized manufacturing, decentralized manufacturing, distributed manufacturing, and point-of-care manufacturing. Those are the 4 different strategies that one can have. Now, I think you might find that some of the newer terminologies like point-of-care manufacturing, the definition might be a little bit different than what you might be you might have understood what that is. And I think that is something we as the field will need to define what that is, particularly for cell and gene therapy products. The definition you will see is developed for all products. But as we always find out in this space, that cell and gene therapies have somewhat uniqueness to it, and it could be even in this how we define what's point of care might be also be the case. So bear with me. So this is centralized manufacturing. I think everybody, almost everybody who understands a little bit of manufacturing probably understand what this is. This has been around all the time. Almost everything started out as centralized manufacturing. What this is that you have a single manufacturing facility that produces and distributes the product to the end user. So it's a traditional way of manufacturing products. What's a little bit challenging for cell and gene therapy products here is that as I mentioned a little bit about the logistical challenges in terms of manufacturing within the facility, perhaps even within the clinic, but definitely between the clinic and the manufacturing facility. That logistic challenge is sometimes a nightmare, especially when it's an autologous product like CAR T cells. And that's where this manufacturing model works to a degree, but hope to have something that works even better. So what came after that was decentralized manufacturing. It's not too different from centralized manufacturing. It's that you have a little bit more manufacturing facilities in different regions throughout the country. It is a little bit closer to the clinical sites, but not right next to it. This is the decentralized manufacturing model. Here, you definitely have the manufacturing agility because the supply chain and it really helps with the supply chain. It shortens the supply chain and then it also gives a lot of reliability by having this kind of model. Here, what's unique about it in this model is that each manufacturing site will have its own quality oversight. So that's something I'd like to point out. But once you start having multiple manufacturing sites, you will have to worry about the comparability, product comparability, and assay comparability between the sites. Let's say you have 3 sites, then you want to make sure, but they're making all supposedly same products, then you want to make sure that they're actually making same products. And the test methods are comparable, so they're measuring the same thing, and the readout is meaningful. Across these facilities. So those are the kind of things that we run into when we have this kind of model. Now, the next one is distributed manufacturing. Here, the difference between this and the previous one, decentralized one, is that it's not that it doesn't have multiple sites, but each of the multiple sites are actually much closer to the clinic. So what really happens here is that usually this kind of setup starts out as a single centralized facility, but that facility decides to deploy same manufacturing units to multiple places. You can think about GMP suite in a pod, or a movable or mobile trailer. They could actually move around from one county to another if they want to, but it's identical setup inside. So those are called distributed manufacturing. Definitely does increase manufacturing agility. Again, it runs into that multiple manufacturing facilities, which has to do with the product and analytical comparability. Here, this model actually has a single centralized quality oversight, unlike the decentralized one. Now, the last model is point of care manufacturing. Here, I think some people might not agree that this is the point of care I was thinking about, and I personally have my own point of care manufacturing model as well, but this is how currently FDA defines it. And what this is that it is the manufacturing occurs at the host site, and it is close in close proximity to the patient care. Again, it has that challenge which has to do with decentralized manufacturing units. But here, the centralized quality oversight is there. The main difference between this one and distributed manufacturing is that in point of care manufacturing, we do not assume that each site is on the GMP environment. It's a non-traditional GMP environment. So what does that all mean? I have to say that the United States and every other country who has regulations would have to stay within the bounds of regulations, regardless of how novel the idea is, until there can be some change to it. And I have to say that for something like point of care manufacturing that we just saw, or even the distributed manufacturing to some degree, we have a challenge in terms of how it's conceptualized, what kind of regulation needs to be followed. So at the moment, all 4 scenarios require full compliance with the current regulation, which requires IND and BLA for each of the products. And in each site, somebody which is a sponsor for the IND or the BLA holder for the BLA will need to be responsible to ensure that everything is followed per regulation, including ensuring that there is GMP compliance in each of the sites. So that's how the regulation is set up for these 4 type of scenarios. I do want to say that for the point of care manufacturing and the distributed manufacturing, which are probably the most interesting manufacturing strategy for cell and gene therapy products, what I presented is our FDA's current thinking based on current regulation. But I do say that there is no other guidance documents or policy being established for this that is out in the public yet, and that's for cell and gene therapy products in specific. But there are a lot of discussions internally going on in the FDA, especially in CIRM in my office, where cell and gene therapies are regulated, to think about different scenarios, different paradigm, and how some of these different paradigms would apply to different types of products that are in a different stage of development. So we have a lot of ifs and what and considerations that we're still having. If nothing has been finalized, but I do want to share that thought with you, that something that we're thinking internally at the FDA. At the moment, we're handling this on a case-by-case basis. And I think this is really a true case where you do have to handle it on a case-by-case basis, at least at the moment. There has been some interactions with some of the product developers who wants to use this approach. And if you're one of those sponsors or product developers or technology developers who wants to further develop something in this area, it is never too early to come and talk to FDA. So I encourage you to also consider it as well. And this is my last slide. And thank you for your attention.
Thank you so much, Dr. Maitland and Dr. O. It was very informative to listen to both of you. I just have been informed by our gracious hosts that in the sake of time and for the people trying to go to the Dodgers game, I guess. I'm not very good. I still marginally follow USC, but don't even ask me about that. That we will both not take questions right now, but our speakers are still here if you have questions. And also invite Dr. Liu Wang to chair the next session without a break.
I had the great privilege of moderating the last session, which is, as we all know, what stands between all of y'all and cars and traffic. And we are running a little bit late, but actually the Dodgers game, I think, starts at like 5:08. So honestly, like you were gonna get stuck in traffic. So just stay and we'll just keep going and it'll be great. And this is definitely gonna be the best session. Because we have Daniela Bota, Svensson, and Mark Walters here talking about the future. Where we're headed next in cell and gene therapies, especially as we sort of look into answers, CIRM's new sort of focus and impact, and their strategic impact of this. So I have everybody's bio, which they're all very lengthy and we could all extremely complicated. I'll probably skip through the interest of time and start with Dr. Whitman.
Thank you so much, Leo. And it is my pleasure today to talk about something that I am very familiar with and not very familiar with. So you're going to have to take your pick. Basically, something that is so new and innovative that articles get published and news get shared every single day. And what we're going to be talking about are the CAR-Ts beyond cancer. And for the people that had children, you remember that's very important to say to infinity and beyond. So that being said, this presentation was also asked to be at the public level. So if anybody gets bored, that's okay. Everybody can check their email. But I want to start very simply with what are CAR-Ts and why to engineer CAR-Ts. And when we think about CAR-Ts, the first C stands for a very interesting word, which if you have studied mythology, it's chimera. And chimera was a very interesting creature that was made out of two different creatures. So the reason to call this a chimeric construct is because it's trying to imitate the characteristics of the two most important immune cells in our body, the B cells and the T cells. So these CAR-Ts are modified T cells, which are able through genetic engineering to have on their surface, in a way, express and on the outside of the cell, an antibody that's going to recognize antigens, foreign antigens, and by connecting the T cells with a foreign antigen, we'll be able to bypass a lot of the steps that are needed in order to activate the T cells and for the T cells to serve their function, to eliminate from circulation or from normal organs things that do not belong there, like cancers, like infections, like even abnormal immune cells that are there on the wrong place and doing the wrong things. So very simple, we have a chimeric cell, a cell that's supposed to do a two-in-one job and do it well. So we are talking about living drugs. You have heard a lot about living drugs. So where are those living drugs coming from? They're alive. They come many times from the patient that we are treating. You see here a very simple drawing of how do we make CAR-Ts in cancer. We have a cancer patient. The patient goes through a process that's called leukapheresis. We isolate T cells. The T cells are genetically transformed. There are different ways to genetically transform them. We propagate the cells. We test them for their activity. And then we re-administer them to the patient in combination with whatever the patient treatment is. The hope with those treatments is, of course, that we will be able to target now specifically and directly a target cell, in this case, a cancer target cell, but it can be any target cell that you want to remove from circulation or from a normal tissue. You have the target antigen binding to the T cells, activation of the domains, and then with the activation of the stimulatory domains, the release of the substances that will create the lysis of the target cells, which are perforins and glycine. So that was very fun. And now we're starting to go on the memory lane. And this is 2017. And in 2017, we had the first CAR-T approval. And the first CAR-T approval was for hematologic malignancies. The product is called Yescarta. And it was doing something very interesting and very simple at the same time. And this is the cancer cell. The cancer cell is actually a transformed B cells. And those cells, majority of normal and transformed B cells, have a receptor which is called CD19. So cells that are now, these cells that are targeting the CD19 were able to remove from the circulation those sick B cells that are creating leukemia. So this is what happened in 2017. And then things started to move. So right now, if you could see, Kymriah and Yescarta, both of them got approved in 2017. And then we have one CAR-T in 2020, two in 2021, and one in 2022. So we are doing very well. We have 6 FDA-approved products. I'm not going to talk about the costs. We already had that. But what I would like to show you are the indications. And if you're not a hematologist, I'm not. The thing that I always look when I look at those diagnoses is how many of them do start with L. So if you look at how many of them start with L, you're going to discover that those treatments, though multiple and targeting a few antigens, not many, and not very specific, are actually able to be treatments and sometimes even cures. They have made a lot of changes. But majority of the treatments are leukemias and lymphomas, which tells you that we have made a lot of progress on hematologic malignancies, but we haven't done similar progress in solid malignancies. And we have not yet made that kind of progress outside of cancer. So, can we do treatments with CAR-Ts for everything that we can think about, from common cold to leg injury? Technically, no. What we need in order to have a condition amenable to CAR-T therapy is, first of all, to have a good target. We have to have a cell population that if we eliminate or at least consistently reduce, we will gonna control or even cure a disease. Now, why are CAR-Ts not moving a lot in solid malignancies? One of the reasons is because solid malignancies are very non-homogeneous. I will talk about what I do, which is glioblastoma. And even in the best case scenario, 20–30% of the cells will express a certain antigen, the rest of them will not. So if you target that antigen, you do not have one specific antigen, and you are not going to be able to remove the percentage of cells that it's needed, with the exception of minority of patients where we're going to have better expression. And then remember, if you have that key cell population and they have your key antigen, also you have to make sure that that antigen is not present on normal cells. Because if you start eliminating the normal cells from circulation, then you are creating a lot of normal organ toxicity. So as you could see, the bar is pretty high in cancer, but it is even higher when we think, as I started to mention, about the environment on which those cells populations exist. And here we have my friend, the solid tumor, the one that I'm still trying to see if we can do something about. And what you mentioned when you look at this solid tumor are a number of problems. There are a lot of mutations on those tumors. They have very high number of mutations, which in a way makes them amenable to being recognized. But when you are trying to recognize with one antigen and you might have 100 different antigens, that is difficult. And then the other problem that I'm not going to discuss a lot about it is majority of the cancers, the cells around the tumor cells are very inhibitory to immune responses. We talk a lot about CAR-T exhaustion and loss of immune responses because a lot of the environment of the tumor works to inhibit the immune response. So now let's look at the other one. That's the non-cancerous disease. We're going to address a few of them. Do you need to remove all the disease cells from a non-cancer disease in order to get improvement? No. Majority of the time, if you eliminate a good population of them, the patient will improve. You don't have a hostile environment. Most of the time you have normal tissue and the mutational burden is usually low or absent. So that makes people think that maybe that we have started with those cells and this therapy, this living drug in cancer, other indications might be even easier to target than cancer. So what can CAR-T potentially target? And when you look at this list, I remember about 20 years ago when we discovered about telomerases, and the list is what we're going to do is maintaining the telomerase length. And the list was about as long as this. But if you look at this list, you should be in awe, but you should also think about what does it really mean if we can find theoretic uses and potentially approvals for CAR-T treatments for all this list. And I know that Dr. O is like, oh my God, right now, because this is a very, very long, comprehensive list for cellular therapy, and maybe he needs to get more people on his office. But actually, if we start with the first column, which are the autoimmune diseases, and we stop before diabetes, and we go to the last column, which are infectious diseases, there is actually work done on those CAR-T protocols, and some of them are showing quite promising results. So though in the middle, I think it's more aspirational, and that's coming from Dr. Baker. So if you don't like the list, I added to it. Please do have the reference so you can connect with them directly. But I just wanted to show you how fast things come. So what you're going to see from now on is that majority of the references that I'll show you are less than 12 months old. So this is what's happening in the last year. And this is, a majority of them are in Nature, Cell, or Science. So probably those people are looking at what they are doing. We trust them. But this is the first notice of the first 2 patients that received CAR-T cell therapy for HIV. And then this was a specific target, the GB120. We are and actually, Merdad, you are interviewed for this article. I don't know if that counts as an authorship in Nature Medicine. We can talk about that later. But definitely that will go on your file as being a public mention of your work. And it's about the hope of being able to treat and cure an incurable disease. I'm not going to talk more about infectious diseases with Merdad here. But I'm going to move to something that hopefully we don't have a lot of specialists in the room. And I'm going to be talking about current CAR-T studies in autoimmune disorders. And if you listen to the public news, and we're going to go through that a little bit more, the changes in our lifestyle and the changes in our environment, for reasons not fully understood, are increasing the incidence and prevalence of autoimmune disorders. In addition to that, there is a high burden of disease and a lot of accumulated disability. And this paper from February 2024, again, was looking at the number of CAR-T studies open at that time in autoimmune diseases and was looking between 7 years from 2017 to 2024. And if you look in 2024, there are 40 studies on those three conditions, which are majority of them, plus some others. I'll mention what the others were, with the lupus being the most prevalent condition, followed by myasthenia and multiple sclerosis. So I'm going to talk a little bit about those three conditions and where we are with those three conditions, because the results are quite interesting. But there are also clinical trials that in that field, some of the ones that I will mention, that we actually either conducted in the UCI Alpha Clinic or we are still conducting in the UCI Alpha Clinic. So this is another list. And what you're going to see here, which is very interesting, is it initially there are very specialized companies running those clinical trials, majority of them on the same platform, in different immune disorders. So we already talked about lupus or SLE and myasthenia gravis, but as you could see, there are other diseases that are coming, like inflammatory myopathy, systemic sclerosis, pemphigus, rheumatoid arthritis, hidradenitis suppurativa. So there are a lot of conditions, all of them having the same backup, which is immune stimulation. And interestingly enough, if you look at the target, you're going to see that CD19, which is the target for the classical CAR T therapies that were originally developed for hematological diseases, is actually the most common target. And the other target that we see, it's the BCMA. I'm going to talk about that later a little bit, but that's again a B cell maturation antigen targeting immune cells. So this is a publication from 2021. It is actually a case report. It's the first case report in New England Journal of Medicine. And it reported on a patient that has received CD19 targeted CAR T, if I remember, was actually Yescarta in refractory systemic lupus. And what you could see on this paper, I just tried to get some interesting data. The CAR T treatment was about here. And what you could see, it's a plethora of symptoms and laboratory abnormalities that this patient was suffering for immediately before the CAR T treatment. And now you could see that after the treatment, those symptoms, including laboratory testings, physical symptoms, skin symptom, arthritis, all of those were reduced to just being positive on blood testing. So this was very promising, and this ended up with more groups trying to ask the same question. And this is where we are with a publication again from the England Journal of Medicine in 2024. This was a combined study. Now we know if it's a basket study or a platform study. We are working on it. I would say it's a basket study for today. And it included patients with three different immune conditions: lupus, inflammatory myositis, and scleroderma. So I'm just looking at lupus here because what I want to show you is that they have treated 8 patients with lupus, a total of 15 patients. And the interesting part is that all 8 patients went in remission. And all the 8 patients were not on immunosuppressive drugs. This is about 6 months after the study completion. And if you want to look at the other patients, not that many of them, like the myositis, the muscles were normal and the patients are not on steroids anymore. So this is quite excellent. But what is also equally interesting and excellent is that they had 1 cytokine release syndrome in 10 patients of a grade 2. And that patient had a grade 1 neurotoxicity syndrome and pneumonia. So those are very positive results because at least on the world that I live on the solid malignancy, the risk of complications with the CAR T therapy, at least the classical one, runs around 30%. So let's go now to myasthenia gravis. And this is the CARDS 08. And this is where we start talking about innovation. Because remember, when we treat cancer, we are looking many times at a little disease and in short intervals of time. Now, when you talk about myasthenia gravis, it's definitely disabled, it's definitely difficult, definitely will end you in the hospital many times, but it's not a deadly disease. So the risk-benefit ratio now is starting to change. And the safety measures have to be, in a way, more drastic and the balance than when we think about cancer. But this is an interesting concept where the cells, instead of having permanent constructs, were transfected with an mRNA. And the therapy is again BCMA, which is the antigen on the plasma cell, plasmablast, and plasmacytoid dendritic cells. And we were part of the study 1B2A as well as the 2B study with Dr. Tassin Mozaffar being the investigator at our site. But what's interesting with this therapy is, of course, this is a short-acting therapy, but you didn't need lymphodepletion. You could administer it outpatient. We ended up administering it kind of inpatient because we wanted to make sure that you really can give CAR-Ts outpatient. You can. And we wanted to make sure that we are following those patients correctly. And there is no risk of genomic integration, at least theoretically. So those patients did quite well on the phase 1, safe and well tolerated. And then the phase 2B was started. And the data from the phase 2B were just presented basically a few days ago. And officially on their website, they're saying that they want to have a end of phase 2 meeting with the FDA by the year end. And they basically mentioned that they're starting to plan their phase 3. The interesting part is that they were seeing significant improvements in majority of the patients. Their responders are doing very well. But what I will go is that they didn't have any CRSs and any ICANS. So something to think about when we think about the cost of therapy, if appropriate. Can we have therapies where you do not need to have a permanent construct, you do not need to do conditioning, and you do not need to keep the patients on suppressive medication? Now, let's move to multiple sclerosis. And this is again starting in June 2024. Was a report of 2 patients that were treated was a CD19 CAR T cell, and this is the Kyverna. Kyverna is one of the companies that's right now very active with the clinical trial portfolio. And the patients did well. The patients actually got CAR T cell expansion in the CSF with no toxicity. And apparently there are no signs of ICANS. So this also was newsworthy. This is the article coming again from in Nature Biotechnology News. And being newsworthy, also something happened. And what happened here makes me quite interested, as I am trying to be an observer in the world of clinical trials, is that the small companies and mid-sized companies that were doing this work before started to be purchased by much larger companies, reminding me of the times, way back day when we were developing biologics. So even though the clinical trial that we are opening right now at UCI, it's done on the Juno Therapeutics platform, it is actually, Juno is now Bristol Myers Squibb company. So we are very excited to do this study. We'll hope that our patients with relapsing, remitting, and progressive forms of multiple sclerosis will benefit of it, because it's, again, a very disabling disease with almost no treatment. And just kind of to show you what we are doing in the autoimmune disease realm at UCI, this is a list. It's probably not the most updated list right now because it keeps changing. We've been involved in a number of the CAR-T studies for myasthenia, for myopathy, the new study for multiple sclerosis, and we have 2 studies in our portfolio for lupus, one on the CAR-Ts and the other one on NKs. So that being said, what are our challenges in outside of cancer? Some of them are old, some of them are new. We still have to worry about toxicity. The fact that we haven't seen it, it doesn't mean it doesn't exist. But we also have to care about, will they persist? Is there such a thing as antigen escape? And how bad are the off-target effect? And then, of course, like always, what's the cost of therapy? Now that we are thinking about the fact that we're going to treat potentially thousands of patients. So this came in Cell, I think just last month. And this is a study from China, where instead of using patient-specific autologous T cells, they use allogeneic, off-the-shelf, healthy donor, CAR-T targeted T cells. So actually, those patients did well. Those patients had also autoimmune disorders. They're a mix of lupus and other conditions. I think one of the patients also had pemphigus. So this might be another way to reduce the costs, even though hospitalizations, conditioning, and everything else will be needed. So what do the future bring? I don't know, but autoimmunity has reached an epidemic levels, and there is a call to action to address it. And if my magic globe tells me correctly, it's a possibility that autoimmune disorders will be the next logical field. And we have 10 million people suffering with it in the United States right now. How are we going to democratize the use of CAR-Ts? I showed you a few examples, and I hope more will be in the future. And while we are talking about this, just wanted to show you guys, I think those 3 windows, our new inpatient cellular therapy unit in the new UCI hospital that's going to open in February, and I'll be happy to invite you as soon as we can look at it from the outside, because of course inside it's going to be very sterile and we're not going to be able to visit. Thank you.
Okay, welcome everybody to see this and my slides up. Great. See JT's still here, so clearly he's not going to the game, otherwise he'd be in the traffic already. So if you get the first slide up, please. So I'm going to talk about, I'm switching gears now, talking about the brain,
Okay.
in the body. And these are my disclosures. I'm focusing on CIRM and what CIRM has been doing in the neurospace. And as all of you know, 1.14 billion has to go to neurological diseases. So I think it's a very important area for CIRM and a very important area that we should be thinking about. So luckily for this talk, just published a review on this whole topic. So I thought I'd just use this review and take you through it. I'm not going to talk about any of our work today. Rather like this previous speaker, just discuss where we are in the field, neurology, and brain disorders. And I have to credit my wife who helped me write this, a long-time colleague. It's a lot of fun to write, a lot of evenings discussing what's here. And I also want to say that the editors actually asked us not to mention independent funding bodies like Fox or NIH or CIRM. This is an overview of the field, and it's an academic review. But today I'm going to actually highlight where CIRM has played a major role. As an overview, there's kind of 3 different sources of tissue for regenerating the brain and doing cell therapy. I'm really going to focus on cell replacement therapy, not putting cells in the spinal cord that modulate like MSCs, but cells that actually go into the central brain tissue or the spinal cord and replace neurons or astrocytes. The 3 options of cells are embryonic stem cells, as you all know, fetal brain tissue, being a big source of cells for brain therapy, brain stem cell therapy. And of course, recently, iPS cells has really transformed our ability to do these kind of studies. Everybody has to go through a stage of a progenitor cell, and then you have to really drive it down to the target. And the 3 types of cells we're most familiar with are astrocytes, neurons, and oligodendrocytes. And of those, the neurons are the ones we focus on. These are the neurons in the brain that you're all using now to use your iPhone or whatever you have to be doing, maybe watching. Those neurons are firing, and most of the neurons were born before you were. So they were actually in your brain at the fetal time, and they've all connected. So the complexity is connecting those correctly. We can get back in the brains. I'll tell you Parkinson's in a moment. And then the other two, the astrocytes and the oligodendrocytes. The oligodendrocytes are very important for things like spinal cord injuries, because they can remyelinate. And the astrocytes are kind of this global cell, like a fibrotic cell in the brain, that can regenerate tissue areas by providing a new source of support. They're an interstitial tissue in the brain that can support neurons, and so these are also very important and vital therapy. And really, it's GMP manufacturing you've heard today has to be done at this first stage. Then the other challenge is putting cells actually in the brain. So that's my kind of global overview. In Parkinson's, I'll run through a few diseases. In Parkinson's, the goal is to replace dopamine neurons in the brain. Those are the ones that die in Parkinson's and give you the bradykinesia, the resting tremor. And it's like an oil in striatum. So just putting the dopamine neurons back, providing the oil, it's going to be effective. And this is really the lead area for neurological cell replacement therapies. Really, it was started by Ulla Lindvall in the 80s. And the first patient was transplanted with fetal dopamine neurons by Ulla in Sweden. Since then, there's been literally 20 or 30 trials. Then it switched to stem cell technologies. And I've listed here the trials currently ongoing or completed. It's an exhaustive list of all the trials that have been done. And perhaps the most exciting is this one at the top here, which is by Blue Rock Therapeutics. And this just read out, it's a press release recently, and they're moving now to a phase 3 safety efficacy trial for this trial. So I think this could be one of the first neural products that could be approved by the FDA if things go well. It's the closest one we have to be approved. CIRM is sponsoring this trial, volunteered by Aspen Neuroscience, and this is an interesting trial. It's an autologous approach. They're actually getting skin cells from the Parkinson's patient, reprogramming it, making dopamine neurons and putting it back into the same patient. They're not using any immunosuppression. It's a fascinating area. And many, many in between. And this is a bit like CAR T. Dopamine neurons for Parkinson's, a bit like CAR T for neuro. There's about 7 or 8 companies or more now getting into this space. They sense and they smell the fact that this is going to get approved. This could be a large therapy. As we heard, there's 1 million people with Parkinson's, and this could translate. And there are complications with Parkinson's. The main problem here is that the neurons that were born down here, in Parkinson's, you have neurons in the base of the brain that project up and release the dopamine up here in the striatum. And what they do in the transplant is they transplant the cells ectopically in the striatum. Now, they're not going back where they should go. They should go back down here in the brain region of the brainstem. The reason they don't put them here is they can't put projections all the way back out to the striatum. So this is called an ectopic transplant. So we're dealing with issues like potentially dyskinesias in the patient. This is dysregulated and not the correct type of correction. So there are definitely challenges. What we'd love to do, and this is dyskinesias, if they're going wrong. But the technology is moving very quickly. What we'd love to be able to do, and the magic here on the screen should happen, is put the dopamine neurons here and make them project back to their correct connections as a phase 3 of Parkinson's. We could do that, then we can restore function and circuitry. That's going to be really exciting. Because now you put the neuron back into the correct circuit for long-term effects. But we might get enough out of this ectopic transplant to regulate the dopamine release and provide symptomatic relief for patients. So we're very excited by all of it. And this neuronal replacement and connections is really a challenge for neuronal replacement across many diseases. And I don't have time today to go into many others, but I will mention epilepsy. Lou Gehrig's disease. This is one I work on most. And here, it's not possible to transplant the motor neurons die in ALS, and you cannot transplant a new motor neuron and get it to the muscle, or put it in the motor cortex and get it to put a growth of axon all the way down to the spine. So we can't do that, unlike in Parkinson's, where you put the dopamine neuron in and get it to release it. And the reason is we control movement, you need to have that whole connection. The strategy here has been using cells to produce interneurons, which can somehow modulate the environment, or produce astrocytes that can, again, modulate the environment and save those dying motor neurons, stop them from degenerating. And there's a number of groups who tried this. These trials are already done, and they had safety effects, but they couldn't get through to the phase 3 efficacy. They ran out of funding. So those ones they made them stop. We're very lucky in CIRM have supported the work that we've been doing now for the last 15 years. And we have 2 ongoing. One CIRM grant completed and the other one ongoing in the cortex. That's trying to see if we can see a signal from the transplanted cells and get safety And so we can then move to an efficacy trial. And we're probably going to want the efficacy to be both upper and lower motor neurons. We're focusing on both areas with astrocytes secreting a growth factor. So we're very excited about that. Spinal cord injury, as you heard earlier on today. This has been probably the first area that was showing really nice positive effects over 15 years ago. And here you're trying to bridge, as I mentioned earlier, the gap, the lesion. There's two different approaches. One is to produce an oligodendrocyte. And in the first trial at the top here, this is these are the neural stem cell trials. The Geron trial here is a trial that started making oligodendrocyte precursors so they could remyelinate the parts of the spinal cord that were damaged in spinal cord injury. And this is a very exciting approach. It was actually pioneered in Irvine. There's some Reeve clinics there, organization initially. And of course, with this one, CIRM played a major role in kicking off all of those studies. Geron CIRM took on this as one of their early projects. And, as Katie is here, it was one of the first patients that you heard from earlier on. And in fact, if you read this very recent update on this trial, in one of these trials, what you require is a long-term follow-up study or LTFU. Remember, these trials are usually a 1 year or 2 year trial clinical protocol. But because it's a gene therapy or cell therapy, you have to follow the patients at least 5 years. The FDA would like you to follow them longer. We'd love to follow them till then, because this product is not going away. It's going to be with the patient the whole lifetime if the cells survive. And so it's very exciting that the outcome of this, if you can have a look at this paper, Lineage Therapeutics took over Geron. And actually, the outcomes is quite good. And some of these patients in the 1 year follow-up are actually doing pretty well with the transplant. So this is incredibly encouraging. Let's talk to Katie earlier on to follow up, keep going back if you're a patient, keep going back to keep getting examined, because these transplants that have been functioning over long periods of time may need a long time to actually have an effect. In stroke, really, there's been quite a few stroke trials, and again, I don't have time to go into detail, but there the idea is to replace interneurons around the penumbra of this damaged area. And the second thing is to increase plasticity. So if you can put a stem cell in that releases factors and takes out bad things, you can get more plasticity in and around the stroke site. And really the leader here is Gary Steinberg, and again, CIRM funded Gary project from embryonic stem cells to make neural progenitors. My good friend and colleague at Stanford for many years, and he's in the middle of a very exciting trial, the product called NR1. We talked to him when we wrote this review. He had a lot more to say about it than Nature Medicine would allow him to write. He's very excited about this trial, so keep an eye on this. This is going to be very, very good. If we can get something improving in stroke, it's a massive problem in the US, and I think Gary's approach is very good, and many other neural regeneration approaches use approaches in stroke that are around the penumbra to increase the plasticity after stroke. So we all know stroke patients can get better if they work out and do a lot of physical therapy. And if you have a cell in there that can regenerate areas of the brain and increase the plasticity, then that's good. And finally, epilepsy and demyelinating disorder. And you just heard about MS and CAR-Ts. There's also some work going on in MS and oligodendrocyte remyelination. Steve Goldman's leading those work at a company called Sana. None of that's in the clinic yet. We just focused on clinical trials. And so only in the clinic are really using neuroprogenitors to modulate inflammation in the CSF. There's 2 papers of cell stem cell research done this. But we're not sure that the cells are really integrating in these cases. They're probably just modulating the CSF. But this trial is probably the last trial I'm going to talk about. It's the most interesting, epilepsy. And again, CIRM funded. I was actually at the meeting where they got this grant. I remember very well. This is based on a fantastic product that makes interneurons in regions of epilepsy, and they've done unilateral transplants, and they've just released the data on the first few patients. And 2 of the first patients actually came off their seizures almost completely. Remember the numbers, but they were going from 50 seizures down to about 2 or 3. Quite remarkable. If that holds up, it's going to be incredibly exciting, because the only other options for these patients is to remove that whole part of the temporal lobe. And you don't want to do that to avoid it. So now you can have this transplant, see what happens. And if it doesn't work, you can go and have a lesion later on. You have that option. The other thing is you can't do bilateral lesions. So if you have bilateral epilepsy, you can't take both lobes out. The patient can't survive. But you can do bilateral stem cell transplants of these GABA neurons And the GABA neurons are like little brakes. They pump out neurotransmitters that block transmission. So they stop the focal area of the epilepsy and help the patient not have these horrible epilepsies. So look out for this company. Thank you again, CIRM. They're called Neurotherapeutics. And I think it's a whole list of areas where CIRM is markedly. So what are the challenges? I've only got a couple more slides. I know it's Friday. The challenge is really immune suppression. In the brain, we're not sure that we even need it. The early fetal transplants from Kurt Fried, he never used immunosuppression at all. The fetal tissue directly into patients, not even prednisolone at the time of surgery. And those patients have a surviving graft 15 years later, no immune suppression. Allogeneic fetal transplants. So I'm not sure we even need suppression, but when you manufacture cells in the dish, it creates all sorts of proteins on the surface. So probably stem cell products will require it. We do a 6-month to 1 year suppression. We're learning about this a lot. We know we have cells surviving in our project 4 years in patients that only have 1 year suppression. So actually in the brain, we get a break. You don't need to suppress more than a year probably, and then you can take a suppression away. Unlike liver, kidney, pancreas. Location, we need new devices to try and hit the target, get better cell delivery. That's something that I'd love to focus on. There's this thing that we put a cell transplant in the brain. The disease from Parkinson's might spread into the transplant. There's some evidence of that, but it seems localized to the periphery of the graft. I think most of them will be okay. It's for life, as I mentioned, not just the protocol. We have to follow these patients consistently. I think this is where the government, the FDA can come in, because if the company goes down, who's looking after the patient? We need to have regulations in place, either the NIH or the federal government, that if a transplant is put into a patient, they should be followed by the government long term, even if the company goes down. We need to traffic this, follow the cells. So most transplants, we don't know where the cells are. If we could have an imaging technique that we could follow whether the cells survived in the spinal cord, okay, brain, stroke patient, that's going to really help us take the field forward, because we're lucky and unlucky in ALS. We get post-mortem tissue, these patients don't live. In most cases in Parkinson's, you've got to see the transplant until the post-mortem, so you don't even know if it's worked or if it's survived. And then finally, it's price right. We had a long discussion about that today. I have a lot to say about that, but I'm not going to say that. In the future, I think hypoimmune cells are coming. We have our own hypoimmune line at Cedars. I know City of Hope is making one, and I think this is where CIRM can really come together. If we have a hypoimmune line, guys, it's going to change the whole field. Andreas Nage's beautiful paper on immune cells, and Sonia Shreffner, who's actually coming part-time to Cedars, so very happy to have her here, has got this beautiful primate study where she basically made immunocloaked cell in islet cells, primary islet cells, put them into the monkey and they survive. So immune cloaking is coming, very exciting, but with great power comes great responsibility. If that cell gets loose and causes a cancer, it's going to put the whole field back, just like gene therapy could stop. This is what's frightening to me about, when I was in Copenhagen 2 weeks ago at the ISSCRB, apparently there's over 1,000 people in the world that have iPS-derived products infused into their bodies around the world. It's quite remarkable. What's also remarkable is there hasn't, where's wood? But there's been no safety concerns yet. We're all worried about tumors, but there hasn't been any yet, which is great. But it's a nice paper of Manchester. If you have a suicide gene, as soon as the cell starts dividing with this product, it dies, it kills the cell. You get that cycle. So this is the way to protect our cell therapies. I think this technology combined with the cloaking is going to allow us to really get it out of the thing. And finally, a fun look at some of the other things. Maybe we need to manufacture cells differently, and I'll end on this. We've sort of been talking about, could we manufacture in microgravity? Because Drew wants to go to space. CBC here is very excited about what space is like. And maybe cells grow differently when they're at microgravity, and the pluripotent iPS cells will respond differently. They kind of stick to the substrate and they respond in gravity. So what if we could grow them in microgravity? We had this nice review recently. If you want to read about things do with stem cells in space. But finally, this is my last mission, and it's great watching astronauts handling bags of tissue that are floating in the cabinet, trying to figure out how to pipette stuff and fix that. Fun. But I will say, we were successful in the last mission to actually make an IPS line on the space station. So we're very excited. At least it went green. It just arrived back last week. The Boeing flight got stuck, so we couldn't get the cells back. We missed our Uber, our SpaceX Uber. But we got them back now, and they're coming back to see us next week. So we'll grow those out. So they'll be the first IPS lines that we're actually made. Okay. Okay, so here's the ideas from my perspective, and I think the new ideas that you mentioned this morning, Pablo, everybody, JT, are in line with the thinking that we have here as well, and many others. So innovative approaches to replace the cells. So these are techniques to get the cells in the brain well. How to get the delivery devices for getting cells in the CNS. Labeling methods so that we can actually, maybe with imaging, show the cells and where they go in the body, and a hyperimmune line, and help the gene pictures of these to create these hyperimmune lines. So I think with that, I will stop, and thank you very much.
Thank you so much. That was brilliant. I just want to take a second to point out that a lot of these clinical trials that people have been talking about are actually already happening in the Alpha Clinics network, which is great. So we've got the HIV CARS trials, both at UCSF and CSD, CFO, the Kyverna is happening at Stanford and other places, and the Neurona trial, obviously, we talked a lot about. We were really fortunate to be able to actually hear from one of the patients who received that therapy last week at Stanford's Consortium discussion. So, batting cleanup. We've got Mark Walters.
Hi, that was amazing. Very tough act to follow. So I won't bother. I'm actually gonna. I was asked to keep this fairly elementary, and I'm not doing that on purpose. Some of these diseases I'm going to describe, I know nothing about, so I kind of made them. I'm not expert on except for one of them on these, and hopefully that's okay. So, and a lot of this is going to be a bit of a review from what we heard this morning. I was asked to discuss the barriers and opportunities in gene therapy, and we heard a lot about that earlier in the day. So, research and development resources are limiting, safety and effectiveness is a challenge to prove. There's a cost and burden to families participating in these trials, and then post-approval, limited availability, accessibility, but the opportunities are mind-boggling. We just heard some really great examples of what we might expect in the future, but for hereditary rare diseases, virtually any genetic defect targeted, but these are typically one-time treatment with potentially curative outcome with a transformative effect on the lives of the person receiving the treatment. And a lot of these disorders are inherited by underserved populations, so it's a way to reach them. So that's the background about, and I'm going to show a few examples and really focus on CRISPR gene editing. So transthyretin is a protein that contributes to amyloidosis. There's a hereditary form of this. It affects about 50,000 people worldwide. Once the amyloid begins to accumulate in the heart, apparently the lifespan is 2 to 6 years. At that point, it also causes neuropathy. And Intellia had this really fairly basic design. It's a lipid nanoparticle shown on the left. It has two components. One is the guide RNA that, like finding the needle in the haystack, delivers the nuclease to the point in the genome that needs to be edited. And the other piece of material is a messenger RNA for the Cas9 nuclease. So the lipid nanoparticle is coated by ApoE that brings it to the hepatocytes. So this is a protein that's made in the liver. And so the way it works is the nanoparticle reaches the surface of the liver, it's carried into the cytoplasm where the messenger RNA is translated into the nuclease, and together with the guide RNA, go into the nuclease, create a double-stranded break in the transthyretin gene, and when the ends of the DNA are brought back together, is done imperfectly, and it essentially makes it impossible for that particular cell to make any more of this protein that contributes to amyloid. So the concept is get the editor in the cell, stop the production of amyloid. And that should be curative if it's done efficiently and safely. And so this was published a few years ago, but it shows really dramatically what happens. And this is just mind-blowing. So the panel on the left shows the effect of increasing the concentration of the guide RNA on the level of editing in blue. So it approaches 90% efficient editing in the hepatocytes. And then the level of the protein transthyretin drops to about 10%. So when patients were treated at 3 different dose levels, so you see the panel on the right, levels of the protein according to the dose level, low, medium, and high. At the highest level, there's a 96% reduction in protein. So pretty simple concept, works really well. Another really rare hereditary disease, angioedemia caused by a defect in the C1 esterase INH, and it leads to this this condition called hereditary angioedema. So when an attack occurs, there's dysregulation of a protein called kallikrein. It leads to leakiness in the blood vessels, and you get swelling. You get swelling everywhere in the intestinal tract, but dangerously in the throat, and blocks off the airway. So patients are at risk to die as asphyxia. So again, it affects very few people, but it's also, the kallikrein is also made in the liver. So here's the same lipid nanoparticle with the same coding with the ApoE. And the only thing that's the messenger RNA for Cas9 is the same. The only thing that's different is the guide RNA. So it's a different sequence, 20 nucleotide RNA molecule. And so it's delivered to the liver in the same way. And the target in this case is kallikrein B1, guide RNA, and complementary to the sequence of the gene. And so this is what happens at 3 different dose levels in 9 patients who were treated, 10 patients. And so the level of the protein decreases at the lowest level to 60%, and then at the highest dose level, 90% reduction. And this translates into the absence of these attacks over time. So at the lowest dose level at the top, there's still a few attacks that occur after dosing, but none after the 112-day time point. And then at the higher doses, really no attacks occurring whatsoever. So really a spectacular result, very precise editing at a very high level, leading to this profound effect. So Mike Lewis, and then one can imagine a platform, if you will, since there's lots of these gene defects that lead to rare hereditary disorders, that once you have created a master file for the editing platform, and all you have to do is change the editors themselves, one can imagine an accelerated pathway to testing and approval. So Mike Lewis sent me this article, and it also uses the CRISPR technology and focuses on the gene responsible for cystic fibrosis. So this is a disorder where there's a defect in getting water and salt out of the bronchial lung lining cells. And when that happens, the mucus, instead of being liquid and expectorating the bacteria to keep one safe from infections like pneumonia, it becomes really thick, and it leads to blockage of the airway, terrible pneumonias in a decrement lifespan. So the problem here isn't knocking out an overexpressed gene. And here it means repairing a defect because there's inadequate production of the protein, this chloride channel protein. So this is an example of how CRISPR can be modified through a process called prime editing. So it uses the same nuclease, but modifies it so it just has a nickase activity, so it creates a single stranded break in the DNA. And then with a prime editing guide RNA, not only directs the nuclease to the location that's targeted for correction, but also an editing template. So with a reverse transcriptase attached to the nuclease, then it's possible to lay down a new template with the correct sequence and to correct the mutation. And now this is not nearly as efficient as the process I just shared, which is simply creating a double-stranded break and bringing the ends back together. And this actually requires replacing the defective sequence. But nonetheless, this has gotten quite efficient. And in this example that I'm sharing, This was an experiment done in bronchial cells from a person with cystic fibrosis. And the panel on the left shows 25% correction using the prime editing reagents that were as shown. And then the middle panel, which is the one I want you to look at, there are three curves. The black one are healthy epithelial cells. And this is what happens when the channel is activated. There's a change in electrical potential. So it goes up, and then it's possible in this experiment then to block the channel with chemicals, and then the potential goes down. So the black is the healthy normal. The dashed line is the cystic fibrosis without any modification. So there's no change in potential. And then the Vertex drugs that are approved, the chaperone drugs that partially correct this problem by successfully bringing the membrane protein to the epithelium, is shown in red. And then the blue are the edited cells. And so this appears to be as good in its clinical effect as the drugs that are currently approved that have the transformative effect in cystic fibrosis. So again, really exciting preclinical work suggesting that this is a technology that will in the future be used quite broadly. And then in my own field, which is in sickle cell disease and thalassemia, so hemoglobin disorders that are hereditary. And this is just a schematic of the hemoglobin molecule that shows that it has these four components that make oxygen soluble and delivers oxygen to our tissues. And when there's a defect in them, and these are the most common hereditary disorders worldwide, lead to anemia, chronic transfusions, and short lifespan. So there's a master repressor of the fetal hemoglobins, and the switch that occurs at birth is depicted on the left. And this is important because when a baby is born who has sickle cell disease or thalassemia, you can do a physical exam, you can even do a blood count, and they look perfectly healthy. In fact, the only way you can make the diagnosis is to look at the type of hemoglobin that they're making. And they're protected from thalassemia and sickle cell disease complications and symptoms. So the idea is, well, why don't we just go back to the way the babies were born, raise the fetal hemoglobin, and that should eliminate the disease. So the discovery of the master repressor of fetal hemoglobin made for a CRISPR target. And there's a way to target this molecule called BCL11A by CRISPR technology. And again, creating an insertion deletion at an enhancer region in the gene, such that the only cells that don't make BCL11A and repress fetal hemoglobin are red cells. So this was developed by Vertex and CRISPR Therapeutics. And this is the outcome. There have been nearly 30 patients treated now in the clinical trial. And this is the level of editing of the intended BCL11A erythroid specific enhancer. And it settles out at about 60 to 80%. And it's durable through 3 years post infusion. This is the next evo treatment of hematopoietic stem cells that are reinfused after myeloablative chemotherapy. So you would think the prediction is, well, the allelic editing is at 60 to 80%. Fetal hemoglobin should be durably induced, and it should have a profound effect on the disease. And in fact, that's exactly what was observed. So this is a swimmer's plot, and what's shown are those same 29 patients where each lane is a patient going from all those blue dots represent painful episodes resulting in hospitalization in gray before the treatment, and then in green after the treatment. So a 94% reduction in hospitalizations related to pain using this CRISPR technology. So that's amazing. It's transformative, and it shows the power of this technology. And so a lot of companies, and both academic and pharma industry companies, got interested in this. So I'm working with a group at the University of California campuses. So is Beam and Graphite Bio. I'm correcting the sickle mutation directly by homology-directed repair. Intellia, Editas, Beam working on changes in the hemoglobin F, the gamma globin promoter to induce fetal hemoglobin. And then the work I just described, pioneered by Sangamo CRISPR Therapeutics. And Here is the current status of these efforts. So Sangoma has dropped out. Intelia deprioritized their project. Editas is seeking an out-license partner. Graphite Bio terminated due to this serious adverse event happening. First patient enrolled. Vertex we know is approved, and the product is called Casgevy. Beam is still has an active clinical trial to induce involvement, and the HDR project by Beam has been prioritized. So this is what's happening in pharma as a consequence of having 2 licensed treatments. And it is emphasized as the importance of CIRM and the alpha clinics and to keep the pipeline active. So the barriers, we've heard a lot about that, about this morning. Like Genia, $3.1 million per dose. Casgevy, a bargain at $2 million per dose. And then the box warning having to do with 2 of the first 7 patients in the Bluebird Bio-sponsored trial developing acute myelogenous leukemia post-infusion in the early stage of their manufacturing program. So, so approved genome therapy for sickle cell disease, readily available donor, no problems related to allogeneic transplantation, which is a big barrier, but we still have ex vivo manufacturing, high-dose busulfan, with this worry about leukemia post-infusion. Infertility is expensive, and honestly, it's inaccessible to most patients today. So the pipeline is the future, and the work needs to be ongoing. We heard briefly this morning about the Rare Disease Innovation Hub at FDA, and the one point I want to really emphasize is the importance to streamline communications with the rare disease community, because this is the reason why we're doing the work, is to create a benefit, and the best proponents we can employ and work with us are those in advocacy and who have the disorder. So that's what I wanted to emphasize. And then in summary, you know, again, I'm just going to repeat this: important to maintain a pipeline of therapies in rare diseases that are not necessarily aligned with current industry profit margins. And that's the importance of the CIRM mission and resources in having academic innovators engaged. The partnership with community-based organizations with patient disease advocates is pivotal progress. The sickle cell disease story is one of them, thalassemia, and a host of others. And then I think that innovation at FDA and CMS over regulatory review and funding has the potential high impact on treatment development and gaining better access. Thanks so much.
Well, I think that all the afternoon sessions were really amazing. It's my pleasure to introduce Dr. Jeff Golden, who's the Executive Vice Dean for Education and Research at CEDARS. We were lucky enough to recruit him from the Brigham, where he was head of the Department of Pathology there. He's a neuropathologist, academic neuropathologist, and I'm delighted to have him give some closing remarks.
Well, first of all, I want to congratulate all the hardcores that are here, still here. And I know some of you are going to have to get to airports, so I'm going to keep my comments short. I haven't been able to see the entire day, but I've gotten to hear a lot of it. And I don't think there's any hyperbole to say that what the Alpha Clinic network is doing is going to change how we take care of patients in the future. It's really having profound impacts in cancer, in immunologic disorders, neurological disorders, hematologic disorders, all these things that you've heard about throughout today, which I think is just a remarkable thing that we've been able to do. Obviously, there's a lot of work still to do, and I think the building of these, a manufacturing network that's able to be able to work together will really help us, and especially as we think about the overcoming the regulatory burdens that may exist. We heard a little bit about that today. To be able to manufacture closer to the patient, be able to do it in a safe way, and be able to deliver so that patients everywhere, independent of their ability to get different places or pay, are able to receive these kinds of therapies. And the Alpha Clinic themselves, I just want to say one thing, the innovation that's coming from them is truly remarkable. The innovation that is going to drive how we deliver healthcare is going to inform the rest of the country and really the rest of the world as this moves forward. So I want to congratulate all of you on the work you're doing. Thank you for all the work you're doing. And I look forward to this continuing. I hope you enjoyed your time here at Cedar-Sinai, and we welcome you back anytime. So thank you and congratulations.